Good morning, everyone, and welcome to Empowering Patients 2025, a Cell and Gene Therapy Summit. I'm Allison Bradbury, and I'm a principal investigator at Nationwide Children's Hospital and assistant professor in the Department of Pediatrics at The Ohio State University. I also chair the ASGCT Patient Outreach Committee. At the top of today's event, we want to give a huge thank you to the sponsors of the event, Eli Lilly and Company, Ultragenyx Pharmaceuticals, Bayer, and Kite Pharma. Your support helps make the ASGCT patient education and outreach efforts possible. We appreciate you spending time with us this morning as you tune in from wherever you may be. Before we begin, I'd like to take a moment to highlight some other upcoming ASGCT offerings. Registration is open for the 28th annual meeting taking place in New Orleans from May 13th to 17th, 2025. You'll see in the resources tab on the right, we have some great patient advocate offerings at this year's meeting that we welcome you to review, including complimentary registration to those that are eligible and exclusive networking opportunities. We are also excited to offer a workshop on the business of advocates advancing CGTs. This half-day workshop on the first day of the annual meeting will explore real-world business models that patient advocacy organizations have pursued and hopes to foster meaningful discussion among attendees to help guide organizations, whether in the startup phase or mature, as they face many decisions on the best business model to align with their goals and strategies to build their revenue and mission. You'll also see a few of our animated patient-friendly videos played before and after the event. This is just a preview of the robust set of patient educational resources we have available on the ASGCT patient education website. All of this is free to use and share. Finally, save the date for our policy summit in Washington, D.C., happening September 25th and 26th, 2025. We'll bring together policymakers, regulators, and other experts to discuss key issues in cell and gene therapy policy. With that, I would like to introduce our esteemed lineup of speakers and the agenda for the first half of this morning. Dr. Paula Cannon, Distinguished Professor at the University of Southern California and the current President of the ASGCT, will kick off with a very warm welcome to our patient and advocate community. This will be followed by the first session with Dr. Stephen Gray, Professor in the Department of Pediatrics at the University of Texas Southwestern Medical Center. Dr. Gray is the co-director of the gene therapy program and will provide an overview of the different gene therapy approaches commonly used in today's pipeline. We will then have a comprehensive set of talks followed by a panel discussion diving into the complexity of clinical trial design and what this means for potential participants. Dr. Raymond Wong, a clinical biochemical geneticist specializing at the Children's Hospital of Orange County, and Dr. Amy Waldman, a pediatric neurologist and the Associate Director of Gene Therapy at the Children's Hospital of Philadelphia, will each give talks to prepare for the discussion. After, they will come together, joined by moderator Sharon King, who is a caregiver and founder of the patient advocate organization Taylor's Tale, and Dr. Heather Lau, a neurologist and adjunct assistant professor at Yale University, for a live panel answering some of the most critical questions among patient communities when it comes to clinical trials. We encourage the audience to ask questions throughout these sessions using the Q&A feature. We will monitor these questions and answer them live after the recording or during the panel discussion. To make the most of these questions, we'll prioritize those that might be more broadly applicable among attendees. I hope that you find some insightful takeaways, and again, thank you for being here.
Hello, everyone, and welcome to our first annual summit on empowering patients, a cell and gene therapy summit. I'm Paula Cannon, and I'm the current president of the American Society of Cell and Gene Therapy, or ASGCT. And as many of you know, ASGCT is the primary professional organization for anyone who has an interest and a role in advancing cell and gene therapies. So in that regard, our members include scientists, clinicians, everybody from trainees to CEOs from both academia and industry. But we also have many hundreds of members who are patients, their caregivers, and their advocates. And those members make critical contributions to our society, helping to drive awareness of cell and gene therapies, and importantly, to educate the scientists and clinicians who are developing cell and gene therapies about what is needed and indeed what would be acceptable. So in that regard, ASGCT is really a big tent because it takes a big team of people to do this, and our patients and their advocates are a very critical component. So let me briefly tell you about the origin of our summit and our goals for it today. So this meeting aligns with an important element of ASGCT's most recent strategic plan, which has put an emphasis on education and communication. So for today's event and tomorrow, we hope that we've listened well and that we'll be providing content to you that meets the educational needs that we've heard about and have been expressed to us by the patient and advocate community. And this is important for the rest of the cell and gene therapy tent because patients and their advocates play such an important role as partners in developing cell and gene therapies. You guys raise funds for research, you collate data about your own conditions, you lobby, you educate us, you educate your own community, and you generously participate in clinical trials. On a personal note, my own research, which is built around the idea of developing a cure for HIV, has been profoundly impacted by the advocacy and yes, sometimes the criticism of people living with HIV who have demanded a role in this process, making good trouble, so to speak. But they've had much to teach me, and the insights I've gained from this community, I think it's made me a little braver and more creative as a scientist. It's certainly given me a sense of urgency, and frankly, it's a major reason I get up and feel inspired and honored to be part of this work. So as I mentioned earlier, this is our inaugural Empowering Patients Cell and Gene Therapy Summit, and we hope to do this every year and to be providing both education and engaging discussions about cell and gene therapies that are relevant to you. So as attendees, you're going to hear from experts in the cell and gene therapy field, as well as patient advocacy group leaders. And there will be opportunities to ask questions and deepen your knowledge in many areas. Some of the key themes today: we'll be hearing about clinical trials and their design. We'll be hearing from panels of patient advocacy groups about the different areas in the pipeline of developing clinical trials for cell and gene therapies. We'll have discussions with FDA leadership and also discussions around therapies that have hopefully only temporarily failed to be commercially advanced. And then also the considerations and challenges for being involved yourself in a clinical trial. So I think it's a great lineup. So I'd like to end by first of all thanking Alison Bradbury and Kimberly Goodspeed, who are the event co-chairs, for all the work they've put into this. The speakers and panelists you're going to hear from, of course, we also thank. And in addition, I'd like to give a shout out to the dedicated team of staff at ASGCT, Margarita Valdez, Lexi Starosta, and Ali Kujawski, for putting their heart and soul into to organize this summit. And finally, to everybody out there online, you, the audience, thank you for doing the hard work of becoming informed, of staying determined and hopeful, and as necessary, of making that good trouble. Because we're in a time of extraordinary progress and extraordinary opportunities, but also some uncertainty. And because of that, we are so thankful to have you with us all as partners in this critical work. So I hope you all enjoy the summit. Goodbye.
Good morning, I'm Stephen Gray. I'm a professor at UT Southwestern Medical Center, and I'm going to try to tackle a pretty broad topic to give an introduction on cell and gene therapy technologies. This is a lot of ground to potentially cover, so I'm going to just acknowledge up front it's going to be a little bit superficial, try to hit on some major themes, challenges, kind of where you would use different technology, and just I guess as a little bit of a disclaimer, I'm going to talk a lot in generalities when I talk about different technologies or different gene and cell therapy approaches. There's almost always exceptions to those general kind of themes that I'm going to cover, and there's a lot of crosstalk between technologies where these are mixed and matched a little bit. So not to make it too confusing, but I'm going to try to go into this and make it as clear as I can in a sort of high view general sense. I do have conflicts of interest that I want to disclose. My lab does develop treatments for rare genetic diseases, and some of those get licensed to companies or I consult. And anyway, there's that. So again, starting at a really high level, focusing on gene therapy, is the promise of gene therapy is to fix a genetic disease at a source. And the idea, the hope, and the dream is that if we fix the DNA, we've solved the problem permanently, and we're not playing whack-a-mole trying to treat all the downstream symptoms, that once we fix the DNA, the cells and the body will kind of correct itself. And there's a really good book that I can point people to if you're interested, that this was published probably at least 10 years ago, called The Forever Fix. And it gives a kind of good introduction and history. Some of it's a little bit dated now, but it's a good read. So, how does gene therapy work in a big sense? Obviously, a lot of you are going to have experience here where patient has a condition, and through some kind of a diagnostic odyssey, it's identified that there's a pathogenic variant in a gene that causes that condition. And our idea here is to design a treatment or drug to restore the gene activity back to normal. And then you deliver that drug back into the patient. And ideally, we want that drug to permanently fix the gene problem. And normally I would put this slide and I would say we're going to deliver the gene back into the body. But when we're talking about the scope of different technologies, there are approaches where we're not actually delivering the gene, we're just delivering something to modify the gene that's present in the patient. All right. So again, kind of talking, I'm going to start at really high level and then kind of break things down and get into the weeds, and just thinking about how your body uses DNA. That, of course, first you have all the DNA out that's on your chromosomes. There's a copy of this DNA in just about every cell in your body. And then, cells will read that DNA to create RNA. And we always call RNA kind of the messenger. It's taking, the message from the DNA, and it's kind of like a temporary step that the RNA hangs around for a limited period of time. And then your cells use that RNA to make protein. And then the protein is really they're the things that are doing the work in the cells. All right. So when DNA goes wrong, I'm giving you basically a sequence here of what your DNA would look like and how it's divided into quote unquote words or into words that your cells would read. And so your cells will see your DNA, and there's 3 billion letters that make up your DNA. And when your cells read the DNA, it divides it into what are called codons, and codons would be like words. And so you have a signal here, like an ATG, that kind of signals the beginning of a sentence, and then you can have a stop codon, for example, TAG, which is basically the period at the end of the sentence. So your cells will see letters strung out like this, and it will know what those are, and it'll be able to read them. So just to, for a conceptual kind of thought, I'm going to translate this into a sentence that we can read that's structured the same way. So it's all the string of 3-letter words that tell a story. And so we can think about this as our gene. So the cat ate the rat and got fat, the end. So we can think about this as like your normal, normal gene sequence. We'll call it wild type, which is what scientists will call like the normal version. So it's a simple sentence, but we can understand it. It makes sense. It tells a story. So we can think about that like a gene. So then you can have changes, single letter changes in that sentence. And these would be variants in your DNA. So instead of the cat ate the rat, it could be the cat ate the bat and got fat, the end, right? So that would be what we'd call a missense variant. And in this case, we're changing rat to bat, the sentence still makes sense. Like, it's different. The story is a little bit different, but the sentence still makes sense. So scientists, clinicians would look at this and they might not know if this is disease causing. So they'll call it a variant of unknown significance, or a VUS. That, okay, it's different, it's not normal, but we don't actually know if it's causing disease. But then you can have something like this: the cat ate the eat and got fat at the end. Now it's a single letter change that's missense. And in this case, we can kind of get the gist of what the sentence is trying to say, but it doesn't quite make sense. And so this might be a pathogenic variant. And then you can have things like this where, let's say the A in and gets changed to an E. So now it's the cat ate the rat end. That we call that a premature stop. So we're missing the end of the sentence. We're missing the end of the story. All the information isn't there. And so this would likely be a pathogenic change. It would be a disease-causing change. And then, you might get into the weeds a little bit about how severe that mutation is, because in this case, the sentence, like, it still says something. It's just missing all the information. Then you can have things where you are missing one letter. So we're missing the A here in 8. And the cell will still read this sentence in order, 3 letters at a time. So if you're missing one letter, everything else gets shifted over. So we'll call that a deletion, resulting in a frame shift. And this would be a very bad variant, a very bad mutation, because you're basically missing almost all the information of the sentence. It doesn't make sense anymore. Or you can have the other side of this, where there's a letter inserted, and it basically does exactly the same thing. It shifts all the letters over, and that disrupts the words where the sentence doesn't make sense anymore. Okay. And then the last thing that I'm going to cover is you could have these changes, like, okay, instead of the cat ate the rat and got fat the end, the cat ate the rat and got fit the end. Okay, so now the sentence actually makes complete sense, but we've changed the meaning. Instead of the cat getting fat, the cat is getting fit. So instead of rats being bad things, all of a sudden rats are really good things, and we're making super cats. And so now the story is changing significantly. So in this case, okay, we're still making the protein. The protein is still functioning, but now it's doing something different. So that could be a missense mutation leading to a dominant or sort of toxic gain of function mutation. And all of these have significance here, because our approach to treating some of these different mutations could be different. Okay, there's also you can talk about RNA splicing, and this gets into other types of therapeutic approaches, where, okay, we have this sentence: the cat ate the rat, the end. I've simplified the sentence here. In reality, that when the genes are in the DNA, you have introns that break up the genes. And so in this case, I've put in intronic region. So we have this inserted DNA sequence that basically breaks up the sentence. And if you were going to read this as is, it doesn't make sense. So when you code that into RNA, then again, the RNA initially doesn't make sense, but the cells know how to deal with these introns. It's part of the natural structure of DNA and RNA, and that intronic region will get sort of cut or spliced out, and then you have spliced messenger RNA that the cell is able to read and make the protein. So if you have problems with the cell of removing those introns, then you can end up with problems where you don't make productive gene products, and you have pathogenic variants and whatnot. So again, there's some different therapies that are targeted really at this RNA splicing step to and sometimes it can be to correct a problem where the splicing isn't happening properly, or sometimes it's trying to improve this splicing. So let's say you have a good copy of the gene, you have a bad copy of the gene. They would try to make this splicing more efficient on the good copy to kind of compensate for the bad copy. All right. So types of gene therapy, this isn't all inclusive, but you kind of have this, gene addition, which is called, gene replacement or gene augmentation. All of these are somewhat the same thing. You're delivering a copy of the gene. Then you can have gene editing, where you're trying to change the gene in its natural location on the chromosome. You can have RNA therapy, where now we're just trying to deliver like the messenger RNA, ex vivo correction of stem cells, CAR T cell therapies, antisense oligonucleotides. So I'm going to briefly cover each of these, but all of these can be combined with different delivery technology. So sometimes you have using engineered viruses to deliver these therapies, or sometimes you have synthetic nanoparticles delivering these. So there's a lot of kind of mix and matching of technologies. And really quick, there's you can divide gene and cell therapy into two different kind of general categories. One is in vivo, or in vivo means inside the body, where you're taking the therapy and like the gene or the vector or whatever, and then you're just injecting that directly into the body, and you're trying to modify cells in the body. Or you can have ex vivo approaches, and this is where typically like you'd take cells out of the body, you'd modify them, and then you'd modify the cells outside of the body, and then you'd infuse those modified cells back into the patient. So in terms of the vector that's delivering the gene, that actually never goes into the patient. It's all done kind of outside in dishes. Okay, so one example of that sort of ex vivo gene therapy is CAR T cell therapy. It stands for chimeric antigen receptor T cell therapy. And so the idea here is that you're taking stem cells, T cells, out of the patient, and then you're genetically modifying those T cells to recognize some signal called an antigen that's on cancer cells. So you're basically reprogramming T cells or immune cells to attack the tumor. And these types of treatments can be highly tailored to specific cancer subtypes. And some of these are where you're taking cells out of a patient, modifying them, putting them back into the patient. Others are they're trying to create sort of stocks of CAR T cells that are available, that are kind of on the shelf, that could be infused into patients. Generally speaking, ex vivo gene therapy approaches are only targeted to blood cells, and these could be immune cells. And so typically like blood disorders, immune disorders, these are generally speaking good targets for ex vivo gene therapy. Usually the genetic manipulations that you're doing result in permanent insertions into the host cell DNA. So these are permanent. Once the changes are made and the cells are infused, then those cells are going to permanently carry that genetic change. And one of the risks of this is that whenever they're modifying the DNA of these cells and they're inserting DNA into the chromosomes of those cells in a dish, if those gene insertions happen in the wrong place, they can actually lead to, they could cause cancer. So you could infuse those cells back in, and then you might have like one of those cells that had, a gene insertion in the wrong place that starts to proliferate, multiply, and essentially lead to something like leukemia. So this has happened in some patients. This has happened in humans, even when the treatment was otherwise successful. So this is just something where the technology is continuing improving, or monitoring is happening to try to avoid this. All right, now I'm going to jump into more of like the in vivo gene therapy, and one of these is ASOs. This has become a very popular therapy approach to treating various genetic mutations. And the idea here is that we're targeting the RNA, we're targeting the messenger. So we're not really making permanent changes with the DNA. The messenger, this is this transient step, and we're targeting the ASO here. And so the ASO is an antisense oligonucleotide, so it basically will bind to the RNA, and it can either degrade that RNA, or it can do things like changing how it's spliced or the stability of it or things like that. Okay, so if we're going to look at the areas where ASOs are used most frequently, of all these different mutation types, it's really, one application is these missense sort of dominant toxic mutations, because they could be targeted straight to this I insertion right here, to knock down or eliminate or destroy the RNA that makes this other sentence. Okay, so they're highly, highly targeted. Or you can also have, as I kind of mentioned before, where you have the RNA that has to undergo splicing to remove the intron, that the ASO could come in here and change how that splicing happens. Okay, so the advantages of ASOs is they're relatively cheap to make compared to the other types of gene therapy. The safety can vary a lot based on sort of the chemistry of these ASOs and the sequence. And there have been toxicity problems around ASOs that was kind of intermittent. It was kind of hit or miss. Some of these would be absolutely fine, and others would run into toxicities, either in preclinical testing or in humans. And I'd say, generally speaking, predicting sort of the toxicity profile at preclinical stages is getting, the design of these is getting better and better. But it's still something where once these get into patients, sometimes there's unanticipated toxicity. So these can be highly customized to specific gene variants. So the ASOs are the technology that's being used a lot in these sort of N of one or N of few treatment approaches. And one of the downsides of this is that these need to be redosed pretty regularly, say every few months. Some of them last longer than others, but the idea here is that they would have a certain half-life. They're only going to hang around, exert an effect for so long. All right, and then RNA therapy. We should all be familiar with this now, because this is the technology used in like the Pfizer COVID vaccine. And this is where you're delivering the RNA. You're sort of bypassing the DNA and delivering the RNA itself. And so one issue is delivering the RNA. I'd say most commonly, this is using like some kind of a synthetic nanoparticle. And this is all transient, because RNA is transient. But the cells would take up the RNA. It will express protein for basically as long as the mRNA stays around, but you're not changing the underlying DNA. So you're not, like, the cells aren't replacing this RNA. It's just using whatever is delivered, making protein while it's around, and then the effect would go away. So we can think about this being very much like gene addition, but it's not permanent. The drug would obviously need to be redosed. And one of the areas where this type of approach can make a lot of sense is around gene editing. So you would deliver the mRNA for the editor, and so the editor is there transiently. It can modify the DNA in the cell, and then the editor is expressed for a short period of time, and then it goes away. So it's kind of like this hit and run approach. So I think that's an area that's probably maturing, that makes a lot of sense for development. And I'm going to mostly wrap up with gene addition. And this is an area where you're basically tackling all these different mutations. Basically, any mutation, all of these that I've got highlighted here, share the same thing, that you're losing a functional copy of the gene. And so if we put back a new copy of the gene, we're kind of bypassing all these different changes that all result in just loss of function of the gene that's in the cell. So the most common way of doing this right now, there are lots of different vectors that can be used to deliver genes. Some of them are synthetic nanoparticles. There's a variety of different modified virus vectors, but the most common is at this point is probably AAV, or adeno-associated virus, where you're using the shell of the virus like a molecular mail truck to deliver the gene. You're pulling out all the virus genes and you're replacing them with the therapeutic gene that you want to deliver. And then you're injecting that into the patient by some route of infusion. So practical limitations and this is really speaking again in very general senses. And for example, like AAV, the gene is being delivered as a mini chromosome. We can think about it like that. So the gene won't be regulated the same way that it is when it's on the chromosome in its natural location. So the treated cells might express less of the gene than normal. So, depending on the gene and its function, expressing less than normal can be beneficial. It could be completely enough, but just realize it might not be 100% back to normal. You can have situations where treated cells might express more of the gene than normal. And again, depending on the gene, there's some genes that are very dose sensitive, where if they're expressed too highly, then it can lead to toxicities. The other is we think about these as being one shot. They're permanent for good or bad. So after the gene is delivered, the DNA is permanently inserted in the target cells. The gene is expressed. It can't be turned off. So you can't stop taking the pill. And that's one of the really important considerations. Also, if we just think about this, in a large sense about what we're doing, you've got, on the order of about 30 trillion cells in the body. And many of I'm just using sort of brain-directed gene therapy trials that many of those are dosing around 1 quadrillion virus particles. So we're about 30 times 30 virus particles for every cell that's in the body. And just to compare this, like your peak virus particles that are inside the human body during a COVID-19 infection is about 100 billion. So we're sticking in about 10,000 times more virus particles into someone's body in a gene therapy than what the amount of virus particles they would have during a COVID infection. So this is not to be taken lightly. And that's why many gene therapy trials include immunosuppression, and it's usually kind of transient immunosuppression just to blunt the immune response, tell the immune system to kind of quiet down, look the other way while we're doing, injecting all of these virus particles. The other thing is, any of these viral vectors, patients typically get immunized pretty effectively against the virus, the viral vector, when we inject them. And so right now we have to assume that redosing isn't an option. And it might be an option in the future. There's a lot of work trying to overcome these immune responses and allow redosing. But generally speaking, anybody that receives like an AAV gene therapy is going to maintain high levels of antibodies against AAV for years and years. And I'll end with just really briefly on gene editing. I think this is a direction that a lot of things are going towards in the future, but there's still a lot of problems with it. The benefits is that it would be permanent. You're basically fixing the DNA in a cell, and then if that cell divides or does anything, it's going to carry that corrected gene with it. And the gene is expressed exactly as normal, so it's in its normal location. It's regulated exactly how it needs to be. The problems are, the editing efficiency might be insufficient. The delivery of the editing machinery is really a big challenge. You could have off-target cuts to DNA in the chromosomes, and also these editors themselves are foreign proteins and they can be immunogenic. All right, so really quick, I'm going to skip through kind of the summary here. I've covered all of this just for the sake of time, but just touch really briefly when we talk about how the government regulates this. I do want to make a point that federal law prohibits the FDA from reviewing any clinical trial applications using human germline cells. And they also prohibit the NIH from funding research in germline cells. So this is just a big no-no. We can treat patients, but we start talking about treating eggs and sperm. This is illegal. And then also there is, the FDA can be pretty savvy here. They have a lot of experience regulating gene and cell therapy products, and they have mechanisms to kind of have accelerated review of these types of products because they can see where they can have a very high impact sometimes. And with that, I want to point to a link here that the ASGCT has of Gene Therapy 101 that will probably cover a lot of these same topics and maybe dive into deeper details on some of the specific approaches. And with that, I thank everybody for your time, and I'm happy to take questions.
Thank you, Dr. Gray, for that very great oversight of the many different tools and technologies we have at our disposal in the field of cell and gene therapy. I know that wasn't an easy task. We do have some questions coming in our live Q&A, so I'm just going to start with some of those. What is the current payload or limit for commonly used gene therapy vectors?
Yeah, and keep in mind when we're talking about the payload size, it's not just the size of the gene, but it's also the size of things like the promoter and other regulatory elements that control the expression of the gene. So AAV is going to be the smallest, depending on sort of the configuration of AAV that you use. It would be either about 2,200 bases or around 4,500 bases. And then you move into things like lentiviral vectors, which are used commonly in ex vivo gene therapy. And I think those get up around 8,000 bases. And then you can get into adenoviral vectors, which have, mostly fallen out of favor, but those get up, over 20,000 bases. And then you can move to other less commonly used viral vectors where it can get much larger than that, such as herpes virus.
Great. We have another question about the infusion differences. So how can you deliver ASOs, RNA therapies, ex vivo, in vivo gene therapy? And part of the question was, for example, metachromatic leukodystrophies gene therapy, the ex vivo lenti gene therapy requires chemotherapy. So can you comment on the differences in infusion?
Yeah, so there's going to be a big difference here when we're talking about in vivo or in the body gene therapy versus ex vivo outside the body gene therapy. And, the ex vivo approaches, which would be like the MLD, is you're taking cells out from the patient, or you might use an unrelated donor actually to modify cells to try to make like an off-the-shelf product. And in those cases, when you're reinfusing those cells, it can be a situation thinking about something like a bone marrow transplant. Like there's a lot of similarities there. And sometimes, like with a bone marrow transplant, you have to infuse, say, chemotherapeutics that basically kill off some of the immune cells in the body and some of these stem cells to basically make room for those new cells to be infused. And then it gives the new cells kind of a selective advantage to replicate and expand and kind of fill that space. So again, I'm talking in generalities, and there's a lot of different ways to do this. But when you're doing ex vivo gene therapy, that's not uncommon, is to kind of have, um, you know, modeling it kind of like a bone marrow transplant. If we're going in vivo, then it's going to depend a lot on what the target is and sort of what the technology is. So if you're treating the eye, then you might have something where you're directly injecting the therapy into the eye. If you're treating the brain, there's a lot of some approaches are IV. Some require injections into the spinal fluid using something like a lumbar puncture, a spinal tap, or things that are even more invasive than that. And one thing to think about is a lot of the gene editing or viral vector mediated gene therapies. These would be designed so that it's one treatment, one injection, and then a lasting impact. And so with those approaches, since they are a one-time injection, they can be designed to be more invasive because it's just the patient has to go through that one time. But if you're doing something like an ASO treatment that has to be administered over and over and over again every few months, then those are really designed, probably the most invasive approach might be like doing a spinal tap. But there's a lot of newer technologies and chemistries with those ASOs that sometimes they can, it's the field is moving now into approaches where those can be administered IV or maybe even subcutaneously, and they can actually penetrate into the brain. So it's all over, but that's kind of a general overview of it.
Absolutely, that's great. I think this one can be addressed pretty quickly about when delivering ASOs, what type of variant must the patient have? The question specifically said, does the patient have to be haploinsufficient? So can you comment on your very nice beginning of your presentation where you talked about like missense mutations? What type of variants can ASOs be amenable for?
Yeah, so it's really two types. Well, it can be broader, but I'd say one of the more common applications would be something where there's a toxic gain of function, and you're trying to knock down or destroy the bad copy of the gene that's actually causing harm. So that's a really common approach. But then it's not just haploinsufficiencies, but it could also be complete loss of function mutations if you can modulate splicing. And so there's an example of like Milasan, which was developed for CLN7 Batten disease, where I think this was a patient that had a, homozygous, loss of function of this gene, but one of the gene variants was a splicing mutation, and so the approach was to target and basically shift that splicing to a correct pattern, and then it reestablished expression of the gene. The other thing would be haploinsufficiencies, where again you're trying to sort of boost the expression of the good copy.
Great. Are there any trials in humans using gene editing in vivo?
There are. They actually don't. I'm not fully up to date on everything, but I think there was a gene editing clinical trial in the eye. There have been gene editing approaches targeting the liver, and I think some of these are viral vector mediated, and some of them I'm not sure if there's been any that are more of nanoparticle delivered, but there are a few examples.
I think the more common are the ex vivo gene editing, where we now do have approved therapies out there, which is really exciting.
That's a good point, and thanks for pointing that out, because doing the gene editing ex vivo is being done.
If AAV is given to a patient, and as such the patient then creates antibodies in response to that AAV, and then a new gene therapy emerges that is better, can a different AAV be used to treat the same patient and avoid the antibodies that are present?
Yeah, and this is something people ask about a lot. And the reality is, when you develop those antibodies against the first AAV, then those antibody levels typically get very, very high. And when you have antibodies against one type of AAV, they do cross-react to some extent to other types of AAV. So I think that the limitation, the block on redosing probably still applies here. There might be some technology innovations that make an AAV that really just avoids all those antibodies, but I haven't seen a really good example where that's been achieved.
Yeah, I think it's an exciting field, trying to do some capsid cloaking or try to do better immune suppression to mitigate this, but it's not quite ready for prime time for redosing with a different AAV. How do you confirm that gene changes in vivo have happened as intended after delivering a gene therapy or gene editing?
Yeah, that's a good question. So sometimes the gene product is secreted and you can detect it like in the blood or other fluids. Other clinical trials, like a lot of the Duchenne muscular dystrophy trials, they actually do a muscle biopsy and they look for what degree of gene expression is happening, and they can monitor that over time. Of course, that's invasive. You're doing a muscle biopsy, but it's doable. Particularly like for brain-directed gene therapies, we're not going to biopsy the brain. So this is one thing. It just depends on the application, depends on the target. There's a lot of clinical trials where we can't do anything to actually prove that the gene is being expressed, except for just looking at downstream biomarkers or downstream, clinical effects of the treatment.
Yeah. We have a good question about the blood-brain barrier and how does this cause problems for AAV-based therapies?
Yeah, the blood-brain barrier is it's an old problem for any drug delivery, and this is why treating brain diseases has been so difficult historically, with any kind of drug development. There's a lot of advancements right now of kind of identifying some different receptors or mechanisms by which we can sort of trick the blood-brain barrier to let things get across. And AAV, like you have AAV9, which is used in the FDA-approved treatment for spinal muscular atrophy, and that uses a naturally occurring AAV that can get across the blood-brain barrier to some extent. It's not very efficient, but to some extent it can. But now there's a lot of engineered versions of AAV that are designed to take advantage of some of these transport mechanisms to get into the brain more efficiently. So this is a really exciting area of gene therapy right now, and it's not just for AAV; it's also for ASOs, and in other nanoparticle delivery to get much more efficient and targeted delivery across the blood-brain barrier. There's some really very recent and very exciting advancements that are happening.
I agree. So there's a good question about disease onset and progression. So how do different variants result in a earlier onset and progression while others have a more attenuated version of disease? So we often hear about our diseases, occur on a spectrum. So why does that happen?
Yeah, I think there's two things. One could be related to that specific gene. And I was giving examples of different types of, mutations or pathogenic variants. And so some of those variants can happen where the gene is, kind of incapacitated, but it still retains some function. And so that could be a milder progression of the disease because you're not completely missing the gene, you're not completely missing the protein, but it doesn't have full function. So that patient might have a slower or milder disease progression than somebody that's just completely missing the gene. So that's one around the gene itself. And of course, the other side of that is all kinds of other genetic modifiers, just our inherent genetic variability. And then there could also be environmental differences, that cause one patient to progress faster than another.
Okay, I'm going to end on one final question. I'm going to kind of combine some common themes that I'm seeing here. So, for a given disease, how do you determine which the best therapy is, and then also perhaps the best capsid and route of administration for a specific therapy?
Wow. So the short, stupid answer is try to find an expert that knows this field and get their feedback, because it's a really steep learning curve, and the field is evolving pretty rapidly. So you may find a review art, like a scientific review article that's a few years old, and it covers this topic really comprehensively, and you think you understand it, but then in the years since then, there's like major technological advancements. So in my talk, I tried to outline as clearly as I could sort of what different types of mutations and then different types of therapies. But it was a tall order trying to cover all that in 20-25 minutes, and I think I just scratched the surface.
Absolutely. I appreciate you attempting to tackle that. And, I really agree that each of these therapies needs to be, disease specific. We need to understand the mechanisms underlying the disease so that we can most efficiently and also safely target our therapy to any given disease. So really understanding that mechanism of disease and finding the experts that do understand the disease and have the model systems is really crucial. So thank you so much, Dr. Gray, for sharing your insights today and to providing this really essential overview of different gene therapy approaches. Next, we will hear from Dr. Raymond Wong, who will review the clinical trial design concepts as a precursor to a panel discussion on the complexity of study design and cell and gene therapy trials.
Okay, thank you.
So it's a pleasure to be here this morning. My name is Dr. Raymond Wong. I am a clinical geneticist as well as a gene therapy clinical trials physician that takes care of patients with lysosomal storage diseases, which are rare inherited conditions. I'm at the Children's Hospital of Orange County, and I just wanted to say thank you for enabling me to participate. So today we're going to go over some intermediate and advanced level clinical trials terminology. And I think many in this audience are already aware of sort of the clinical trial process. Many of you are already aware of sort of the different phases and why the clinical trials are being done. And so I'm here to introduce you to a bit more advanced level terminology. So we already know that clinical trials are studying investigational medications. Initially, the idea is to see if they are safe, if they cause adverse events, and if they're tolerable in the sense that the way that the drug is being given is safe and does not cause issues as well, because many of the treatments aren't orally administered. They're either intravenously, intrathecally, or somehow into the central nervous system, or even as a stem cell transplant. So let's get on to the good stuff. Ultimately, the idea of any clinical trial, especially phase 3 and above, is to assess whether or not that investigational medicine actually works, and that's a term called efficacy. And even though a study can be phase 1 or 2, especially for rare diseases, the people who are running the study are trying to get a hint at whether or not that medicine actually works or not, even though they're really gauged to look at safety and tolerability. So how we assess this efficacy is a bit more of a complicated conversation. That's what I'm going to be spending the bulk of today's talk on. And really your goal is to get your investigational medicine approved so that all patients with that condition can receive the therapy. So I want to talk about a clinical endpoint. That is the ultimate outcome of the clinical trial. What is that medicine supposed to treat? What is it supposed to prevent? And the emphasis here is on clinical. So I have a few examples here. If you have a medicine for diabetes, you want to see whether that medicine prevents death. for high blood pressure, whether it prevents stroke, high cholesterol, heart attack. Those are all measurable, tangible events that happen to patients. There are also surrogate endpoints, and that's really a biomarker or something that's measured. It can be a radiographic biomarker, it can be a lab biomarker, which possibly correlates with a clinical endpoint. And I underline possibly, mostly because there's a potential connection, but especially in the field of rare disease, that connection is not 100% sure. I mean, even for the exact same conditions, you look at something like hemoglobin A1C for diabetes, the actual blood pressure for high blood pressure, and your bad cholesterol. And these, over many, many years and many tens of thousands of patients, have been pretty much conclusively shown to be associated as predictors of these ultimate clinical endpoints. But the story of that is, again, not quite so clear for rare diseases, and we'll revisit this later. We also have something called a comparison or a control group, and these are the people with the condition who aren't receiving investigational medicine, right? Because if you're wanting to see whether or not your medicine works, you need to compare the people who receive the medicine with the people who don't to see if there's a difference or a benefit. Now, you can have a placebo group, and I think most of you are aware of this, that these are people who are enrolled in the study but don't get the medicine. Or you can have a historical control group where you have a group of people in the past, anywhere from 5 to 10 to maybe even more years ago, who maybe had that clinical or surrogate endpoint measured. And really, there are issues with both of these control groups, which I'm also going to revisit in the next slide. So I'm going to get a little technical here, and I'm going to walk you through this. We're running a clinical trial. This is a hypothetical outcome measure, and it's a number. And the worse it is, the higher it is, the worse off the patient is. And so at the beginning of the study, you have a control group, you have your treatment group. This is really what you want to see, right? Over time, as the study progresses and eventually reaches the end, the marker worsens in the control group and improves in the treatment group. And here, we added some fancy schmancy error bars. And essentially, when you see that the control group is very separate from the treatment group, that's a sign of success. Your treatment worked. You can even add some fancy statistics to show that, this group here in the control at the study end is statistically worse than the baseline. You can show that maybe the treatment group is unchanged. And then you can show a comparison between control and treatment that shows that these two are really, truly significantly separate. And then you celebrate. However, the problem is when you have a clinical trial where, maybe the control worsens and the treatment maybe improves, but you can see these error bars are really wide and you can't really make any, clear statistical conclusions, that's where you start to sweat a little bit. And so the take-home point of these sort of figures is that you really have to pick, number one, a really good endpoint, and number two, a really good control group in order for your therapy to have a chance at passing the efficacy mark and succeeding in getting approved. There are some additional complexities in how we design a clinical trial for rare diseases, as if that wasn't simple enough already. There are a limited number of people globally with the condition. That's just the definition of a rare disease. But there's an even more limited number of people with the condition who will satisfy the inclusion-exclusion criteria for the study. So you're taking a small pie, and then you're even taking a smaller piece, and not everybody may necessarily live in the state or the city or even the country where the clinical trial is taking place. So that gets difficult. There's also this concept of disease heterogeneity. So each person with the condition really has their own degree of symptom onset, severity, how quickly the disease progresses. And if you have a condition that's really heterogeneous, that makes sort of the measurement of a clinical trial outcome even more difficult. And so you really have to select the best study endpoint for assessment of efficacy. And I place that in red because there's considerations over sort of heterogeneity. So you can see everybody kind of clusters around this 4 mark, which is the mean. But you can see over here, it's got the exact same mean, but a much wider variability. It'd be ideal to pick some kind of clinical endpoint that's got less variability. Also, the other consideration is time, right? So if that outcome ends up popping up like years and years from the time of diagnosis, you may need to wait years and years for your clinical trial to get the results. And so it has to be timely, it has to be reasonable, and it has to be as homogeneous as possible. So does that endpoint really truly track the disease? And does it change enough during the study, as we mentioned before? So obviously, for a rare disease, when there's a clinical trial that emerges, everybody wants to enroll, right? That's their best hope, best chance at maybe doing something for their child, for their loved one, for themselves. But not everybody's going to be able to get enrolled because you need to enroll enough people to be able to see a difference. You can't enroll too many people because, unfortunately, there are costs associated with clinical trials. And at the same time, you can't enroll insufficient people that you can't see a difference. So these are pretty significant considerations as you design a clinical trial. There are also ethics. And we kind of alluded to this previously. Having a placebo group for a condition that is progressive and or fatal is really not ethical. To ask somebody to not go on treatment and watch as their disease progresses is just not something that we can stomach. So if you look at a historical control or maybe a natural history study, the issue there is if you are taking data that is reported from a patient maybe 5 or 10, 15 years ago, general medical care has improved and advanced enough that these patients may be surviving longer, doing better, and it has nothing to do with your investigational medicine. It's just general medical care is improving. So we have to take that into consideration. A possible way to get around this placebo group issue is something called a crossover study. It's where you enroll 2 groups of patients. One group receives the treatment immediately and over time gets assessed. And at the same time, there is a group that initially doesn't receive the treatment, and then after some period of time, then gets the treatment and gets assessed. And you can basically look during this duration of time after the initial placebo group gets the treatment to see if they actually improve. But, there are issues, again, if you have a progressive condition, may it have progressed too far for the treatment to have any kind of efficacy. Finally, another ethical consideration for a clinical trial is that many of these phase 1, 2, safety tolerability studies are looking at something called dose escalation. The FDA, at least in the United States, wants patients to start off at the lowest possible dose because they want to look at safety. But the problem with the lowest dose is that you're not sure if that's enough of a dose to actually be effective. And as subsequent patients receive increasing doses of study medication, there may be an increased risk of adverse events. And especially if you're dealing with gene therapy, oftentimes that one dose is your one shot. So once you've reached the low or received the lowest dose, you won't get another opportunity to receive that again. So I think this is my last slide. We really need to get to the finish line. For the US, that means an FDA-approved BLA application, and there are different routes eventually to get to that BLA. But I will say that recently the FDA has been more willing to consider something called accelerated approval, where an investigational therapy is approved on a surrogate endpoint. Lately, this has been more positively regarded. In the past, the FDA really wanted to see clinical endpoints like survival, cognition, various things that take a really long time to develop. After the approval, assuming we get to that point, those treated patients must be monitored for a very long time. And if the clinical endpoint continues to demonstrate efficacy, that approval will continue. If it doesn't translate into clinical efficacy, then that approval will be taken back. So I just want to sum up, this is a really exciting but also a very nervous time for rare disease therapy. This is one of the reasons I went into medicine. Investigational treatments for previously conditions thought to be incurable are being developed. And now with the FDA considering surrogate endpoints, the pathway to approval is easier than even 5 years ago. There are significant challenges, but we didn't go into this field because it was easy. And I am super optimistic for the future that the treatment of conditions, maybe even in the near future, and maybe not even in 5-10 years from now, is going to be very different from the way things are. So I just wanted to thank you all for your time and look forward to taking your questions.
Hi, everyone. I'm Amy Waldman from the Children's Hospital of Philadelphia, and I wear a number of different hats. I have studied rare diseases my entire career, starting with rare diseases such as pediatric multiple sclerosis, which is not, multiple sclerosis is not rare, but pediatric multiple sclerosis and some of the neuroinflammatory diseases, in fact, are. I then extended my rare disease journey to leukodystrophies and even to gene therapy. So I have a lot of different experience in clinical trials, and I'm delighted to speak with you today about why clinical trials for rare diseases are somewhat different. So I know you have just heard about clinical trial design, and so I'm just going to maybe highlight a few things that I think about in terms of clinical trials. And then I really want to talk today a little bit about the reality of clinical trials and some real world examples, some comments that have stuck with me throughout my career that I wanted to share with you. As you just heard, designing a clinical trial is complicated. It begins with understanding the disease and the natural history. We have to know what to expect in a disorder, to know how to modify that or change it over time. It is a very long process to develop a clinical trial. Some people say 10 years, often longer, from the time that they do the animal studies to testing the safety prior to it ever becoming a drug for humans. And then the human trials are often several years, and then the review process takes even longer than that. And as you just heard in the prior talks, there are ethical standards that consider the risks and benefits. It's a whole different language talking about the clinical trials. And these trials are designed with a very rigorous design that is inflexible on purpose, right? Strict rules are decided before a trial begins about who gets the treatment. And so it's not possible, as we do sometimes in clinical care, to make exceptions or to bend the rules, right? If you meet the criteria, you meet them. And if you don't, you don't, which can be a little bit difficult for sometimes people to accept when you're so desperate to have a new treatment. You have to follow a very specific and very detailed protocol. So sometimes these are very consistent with clinical care, and sometimes they're a lot different than what you would do clinically. So there's a detailed protocol or recipe of what needs to get done. And interesting, most of my comments today are going to be about rare disease trials. And what I mean by that are smaller numbers, and we're going to talk a little bit about numbers of patients in a few minutes. There may be a placebo. So for example, some small molecules or antisense oligonucleotide, those trials have in the past involved a placebo arm. Even gene therapies have at times involved a placebo arm. Other trials, a placebo is considered unethical, and other trials, at least in the beginning, as you probably just heard, might be open label, meaning you know that everybody gets drug. But one of the tenets of science and medicine is to do a very careful, detailed trial and randomization, deciding randomly, it's actually not a decision, it's a selection by a computer selecting who gets drug and who doesn't get drug in these placebo-controlled trials is a very important way for us to eliminate bias in clinical trials. So sometimes there is a comparison group and sometimes there is not. And so what I first recommend is that everyone familiarize themselves with clinical trial design and what studies are available for your particular disease of interest through clinicaltrials.gov, which is the website that all these clinical trials are registered through. Now, I can't talk about clinical trials without talking about this one comment that one of the sponsors, a pharmaceutical executive, once said to me. Sometimes the drug fails, but sometimes we fail the drug. So what's incredibly important in any disease is to determine what you're measuring and how you're measuring it. And so we need to understand one key thing as to whether a drug is working or not. So if in a rare disease, how do you know, well, that drug is making a difference in that disorder? And this is not unique to a rare disease clinical trial, but in rare diseases, it's sometimes hard to pick one thing that is going to be uniform throughout the patients to say, we know that a change in this symptom means that the patient is in fact getting better. And interestingly, a lot of our rare disorders, we think more about trying to prevent a patient from getting worse, and we may not be able to show that a patient is in fact improving. So interesting in how we design these trials to try to figure out, is there a measurement that could be applicable to all the patients to really determine whether the drug is beneficial or not. And it's very hard sometimes to compare patients of different ages. You know, some of our rare diseases affect children when they're very young, but also adults, and how do you compare them? And then also comparing people with different abilities, patients that have learned to walk or patients that cannot walk, and how do you say a drug is working in those different scenarios? Okay. Clinical trials are research studies. They are not clinical care. And a lot of my comments today are going to be focused on trials that are typically first in human. So a drug that has never been studied before that now is being given to patients with the disease. So the drug is unproven. We don't have safety data and we don't have efficacy data. It's a little bit different if the drug has been studied in an adult population and now is being given to children, because then we have some data. But in rare disease, we're going to start with a brand new drug that has never been trialed before. So these clinical trials involve risks compared to clinical care, right? It's a new drug. We don't have safety data. We know things might happen to the patient. So this is brand new. And institutional review boards must approve the protocol at each site, even if the FDA has said it's okay to go forward. And interestingly, even though the FDA might approve a clinical trial, a lot of times an IRB or an institution review board still has additional parameters or additional questions, or may not let the study, for example, enroll all ages. They may ask that older patients be enrolled first before giving it to younger patients. So sometimes the IRB has their own suggestions or criteria, even if an FDA has already approved a trial. So that's an interesting added point, that patients who say, well, the FDA has approved this, doesn't always mean that every institutional review board is going to allow the study to be conducted in the same way. And a good example is this age-related one, where even if the trial is able to, or the drug is able to be tested in all ages, sometimes we've been encountered with the IRB where they have asked for older patients to be given the drug first. This point is incredibly important. So rare disease, there's not a lot of patients with that disorder. There may be 2 or 5 or 10 patients in the country, or maybe 30. But the goal is to enroll the fewest number of patients to obtain the required data to prove the safety and efficacy. So we need to enroll the fewest number of patients because, again, the goal is to minimize risk to patients. So even if there's only 30 patients in the US with a rare disorder, not all 30 of them are going to be eligible for the trial. You might do a clinical trial where you start off with 10 patients, not all 30. So not every patient is going to get into a trial, even for a rare disease. So with that in mind, we have to think about, okay, well, who is the best patient or who is the best candidate for a clinical trial? So as I mentioned before, all subjects must meet predetermined inclusion criteria. And there's also going to be some exclusion criteria that must be followed. So there's a list. Sometimes it involves genetics, sometimes it involves labs. The exclusion criteria might involve having tried an experimental study in the past. So there are inclusion and exclusion criteria that must be followed. And that's pretty standard for most clinical trials. But this is one that I wanted to share. The best candidate for a clinical trial is a patient that has medically, nutritionally, and emotionally been stable prior to enrollment. So the sickest patient is not necessarily the best clinical trial candidate. And this is a little bit different from how we think about maybe some of our oncology or cancer trials. So a lot of times in cancer, these clinical trials become available for a patient that has failed a clinical treatment or 2 clinical treatments. And so these have very refractory cancers, and then they're given an experimental drug. That's not necessarily the case for many of these rare diseases. We want to know, is the drug making an impact on the natural history of the disease? So some of the patients that are the sickest may not be the best candidates because we may not be able to measure change, or we may not know what's related to drug and what's related to the illness. So unfortunately, some of the sickest patients are not the ones that are strong candidates for an experimental therapy. Now, some of the pharmaceutical companies will differ. Some people will say you need to be stable for 3 months. Some people say you need to be stable for 1 month. But what they mean by stability is no drug changes, no surgeries, no other medical events. So trying to, for example, a seizure med needs to be stable for 3 months prior to enrollment. And again, each protocol is going to have a different definition of how much time they want someone to be clinically stable. But again, it's not the sickest patient, it's the most stable patient that is typically a candidate for a clinical trial. Now, I will further say the best candidate for a clinical trial is a patient and family that will comply with the protocol and study procedures. Clinical trials are intense. There is a lot of extra testing and monitoring and phone calls and visits to the hospital site. So there's a lot of extra work that needs to be done as part of the trial. So for example, you might have to come to the clinical site every month or every 3 months, but there might be phone calls on the off weeks that you're not there, or there might be some additional lab work that needs to be done locally. So the best participant in a clinical trial is the family that is going to comply with the protocol and study procedures. So for example, this is a patient or family who is committed to the clinical trial schedule, that does not have missed appointments, that arrives early, especially the day before. So for some of these trials, if sedation is involved, right? If something needs to be delivered intrathecally, which through a spinal tap or through a procedure, we really need the family to arrive early, perhaps a day or 2 before, because if there's a flight cancellation or if there is a delay, it is very hard for the clinical team to rearrange the sedation and the time slot. A lot of planning goes into these clinical trials. Provider availability. A provider might be available on a Wednesday, but they're in clinic all day on a Thursday, so they don't have flexibility. So I encourage families, you have to come early, especially if flights and travel are involved. And you have to be easily reachable by phone and email to engage, and text to engage with the study team. If something changes, we need to be able to reach you. If there's a delay or something gets moved up, we need some patients that are going to be actively engaged with our teams. But I want you to think about these schedules. So the schedule affects the patient and caregivers, but also will affect a parent's ability to work for a few days, especially if you have to fly to a clinical site. There may be other children or other responsibilities for family members or pets that are involved. So there are often additional costs to a family, even if the study is paying for travel costs. There are additional costs in terms of babysitters or pet sitters or other things that might factor into a family's decision about whether or not it's feasible to participate in a clinical trial. And the last thing that I will mention is the schedule is determined by the protocol, not the patient, right? So if the protocol says this is the time window, you need to come between this date and this date, we don't have flexibility in that. Like, those are hard set by the protocol. We need the families to travel on the dates that we ask them to travel. This next portion of the talk is probably the most difficult, but I want to share real world experience in terms of participating in a clinical trial. So for a family that's considering a clinical trial, the question is, what amount of risk are you willing to accept, and for what benefit? Especially when we may not be able to tell you if there's going to be any benefit to your child participating in a clinical trial. If it's a first in human study, and we have no idea if the drug is going to work or not, there's a lot of risk without any promised benefit because we don't know if the drug works. So we're going to start with this question: what amount of risk are you willing to accept? And again, going back to the question, who is the best candidate for a clinical trial? Being that the most important factor is really a family that's going to be able to comply with the protocol and study procedures, right? If this is a 2-year trial and you have to come back to the study site repeatedly for a drug or for a dosing or for a procedure, I just want to pause for a minute and say, imagine the following occurs. So thinking about the what ifs to decide if this is right for you. So what if there is an adverse event or a side effect in another patient during the trial? So you're coming to the study site, and now that you hear from the study team and you have a new consent form that you need to sign, even though you already signed a consent form in the beginning of the trial, there's a new side effect. Let's just say it's liver failure. So patients who have been getting this drug, there has been a risk of liver failure. And I'm, of course, making this up. But so now you're enrolled in the trial. Are you going to feel comfortable? Are you going to feel comfortable with your child enrolled in the trial, knowing that somebody else has gotten liver failure, right? When you're committing to the trial, and this is what this first bullet point suggests, if it's a 2-year trial, you're committing to those 2 years. This isn't a clinical care where you're going to try it out and see what happens. And then if somebody else has liver failure, you're going to be like, you know what? I don't really want to do this anymore. I'm going to back out. It is possible to withdraw from a clinical trial, but it has a huge impact on the enrollment, on the study, on the ability to take a spot from somebody else. So if you think very carefully, well, you know what, if I'm in this trial and somebody else gets liver failure and I don't know that I want to continue, maybe a clinical trial is not the right decision for you or your family. What if you're the one, or your loved one is the one with liver failure? So let's say you develop liver failure, and at this point, the sponsor and the medical teams who are evaluating your child says, you know what? We don't know if it's related to the study drug or procedure. It might be, but it might not be. Are you going to feel comfortable as a parent or for yourself coming back and getting study drug over and over again if there's uncertainty about whether the study drug contributed to any of that liver failure? Right? So is that something that is that a risk that you're going to be comfortable with? What if another patient dies during the trial? and this sadly has happened, are you going to feel comfortable continuing in the trial knowing that somebody else has died from a side effect or from the disease? Sometimes you can't really tell. So if somebody else has died after exposure, are you going to feel comfortable continuing in the trial? And if the answer to any of these questions is no, then a clinical trial might not be the right fit for you. Now, I will say, if the death is related to study drug, if the side effects are related to study drug, there are procedures in place in a clinical trial to protect patient safety. There are special data safety monitoring boards that can weigh in. But in clinical trials, it often takes a long time to figure out, is a side effect related to the drug or is it not? So it might take weeks, it might take months, it might take special reviews. And while the sponsor and the IRB and the FDA are all trying to make sure patients are safe, there is a process, and it's not always possible to know whether or not something is related to the study drug, to the underlying disease, or if it was random, right? Certainly, if heaven forbid there's a car accident and somebody gets hurt, that's obviously, for the most part, not related to the study drug or procedures. But if someone, for example, going back to this liver failure example, if someone develops liver failure, it's not always possible to know whether liver failure was related or unrelated. So advice that we have for families: talk to your family and your support system about the logistic, scheduling, and financial about participating in a trial. If you're interested, then demonstrate that interest. So contact the Rare Disease Foundations, contact the sponsor, contact the study site. But just because someone is eligible does not guarantee them a spot in the trial, right? Remember, rare diseases, they're still rare spots in a clinical trial. And please be patient with the study teams and sponsor. There's a lot of work and logistics in terms of going through these trials. A couple more comments I will say: study teams are under confidentiality agreements, and sponsors cannot share information as it impacts the integrity of the trial. So please keep this in mind. It's very frustrating that we can't share certain aspects, but it's to protect the patients and to protect the data integrity. I also want to remind everyone the dangers of social media. I'm showing real data from a clinical trial that has been published. And I'm not showing the videos that are associated with this data, but there have been some videos that say, so-and-so was the second patient ever to receive this gene therapy. And so you can identify the second patient ever to receive the gene therapy, this patient right here in blue. And the sibling was also enrolled, and we can get information on the sibling. Now, one thing you'll notice is this patient number 2, was the best patient in the trial. They had the best outcomes. But on social media, there were some very frustrating comments that were made about the disease and about how this patient, who presumably had the best outcome based on the data, having a lot of frustration about the underlying disease. So I just remind everyone that social media can bring a lot of attention to, if this patient's doing so well, why are these comments so disheartening, right? And there's a lot of unblinding that can happen by saying things like, I'm the second patient ever to get this drug. And I didn't put the identity in this talk, but we can very quickly learn by name who this patient is. So I'm going to skip through this, but my personal ask is that any patient involved in a clinical trial refrains from posting on social media. That's what I ask my patients. It's not a rule, it's not an inclusion criteria, but there's also a lot of patients that can't get in the trial, or are too sick to get in the trial, or wish they could be in the trial, and for whatever reason can't. And I just think it's not appropriate to be posting on social media. So in conclusion, I just want to talk about some lessons learned. Rare diseases have few patients, but few patients also get to participate in these trials. So stable patients are the ones that are prioritized. The commitment to the schedule is one of the most important factors. Risks are real, and I hope I gave you some things to think about today in terms of those what if questions. No individual or personal social media posting. I mean, really, this is a contract between you, your medical team, the pharmaceutical company, and the sponsor to maintain confidentiality. Trials are expensive versus very emotionally taxing as well as financially. And please support each other. In rare diseases, there tends to be some jealousy if somebody gets into the trial and another person does not. But even those patients who are lucky enough to get into the trial do face a lot of really tough decisions. And so please support each other. That's all for today, and I'm happy to take any questions.
Good morning. I'm Sharon King. I'm founder of Taylor's Tale, and I'm senior lead of advocacy at Aldevron. It's a real privilege to moderate this session this morning on the complexity of study design in cell and gene therapy. And I'm joined this morning by Dr. Heather Lau, who's the adjunct professor at Yale University, Dr. Amy Waldman, who you just heard from, medical director of the Leukodystrophy Center of Excellence and a pediatric neurologist at the Children's Hospital of Philadelphia, and Dr. Raymond Wong, who's the director of the Campbell Foundation of Caring Multidisciplinary Lysosomal Disorder Program at the Children's Hospital of Orange County. Welcome, panelists. And I also want to thank our audience for joining us this morning for this discussion. So we've had the pleasure of viewing some outstanding recorded sessions this morning, thanks to each of you. And we've received a lot of questions in the chat, but panelists, I'm going to be directing questions to each of you. I hope that you will feel free to jump in with your thoughts and ideas so that we can make this a really lively discussion. And also, I'll be filling in with some of the questions from our audience as well. So let's get started. And let's begin by discussing clinical trials and that balance between hope and safety. Dr. Lau, I'm going to direct this first question to you. For patients and families, clinical trials often represent hope. However, researchers focus on assessing safety and efficacy, not a guarantee of treatment. Despite significant efficacy over the past 10 years or so, safety still remains a big concern. How do you navigate this difference in perspective between families and researchers?
Thank you, Sharon. It's a pleasure to be here. And just a little bit more about my background. So for the past 15 years, similar to Dr. Woolman, I've worked in rare disease. I trained and was the director of lysosomal storage at NYU for many years, and then the director of neurogenetics, assistant director. Now I'm on the other side in industry, joined 4 years ago in drug development. And today I'm speaking on my own. So with that experience, I think it's really important that we have a frank discussion when we're talking with our patients and potentially either recruiting for a trial. We really need to understand and have a proper informed consent to manage those expectations. So to come to this, that, we do embark on a phase 1 trial with the hope, right, of investigators, sponsors. We do hope that we see some type of efficacy based on our preclinical studies, right? There is some inkling that this particular therapy hopefully will modify that disease. So we are going in together with that hope, but we do need to have a frank discussion with families and caregivers about the expectations. So depending on the phase that you heard about from Dr. Wong and Dr. Waldman, depending on the phase, we may not have a clear understanding of efficacy. And so we have to couch that discussion with that phase of that trial. So for example, in a phase 1 trial, we really need to highlight the unknown benefit with the risk or the unknown risk. So they're both unknown. And so first and foremost in medicine, right, is our motto is to first do no harm. Right. And so that's where we have to meet those families and say, listen, we want to help your child or yourself, but we need to make sure that we mitigate any risk and go into this very open and honest. And so that's how we start to manage that well before they sign up for a study. And in fact, as a prior, when I was not even the principal investigator, but maybe I was the referring physician to another clinical study, that was part of my rare disease clinic discussion. Anytime we think about a potential therapy, well before they go to that site, what are the expectations? So it's, again, having that conversation early and understanding the phase. Now, once you get to phase 3, you might have a sign of efficacy emerging. And so you can have a little bit more of hope with them. But still, I've been through a phase 1, 2, 3 that ultimately failed, right? So really it's about educating like we're doing today, and talking to them about but really pushing with them that process safe before we can move forward. I'll pause there.
Yeah, would anyone else like to speak to that question? Very good. So, Dr. Waldman, my daughter participated in a novel clinical trial more than 15 years ago. And given our experience, I really appreciated your comments earlier about what makes the best candidate for a study. And as a parent, I know that you always want as much information as possible. I mean, after all, this is your child. So when is there when there is information that you can't share, how do you explain this to families? How do you frame those reasons for not being able to discuss certain topics, things that families really want to know, but it's just not possible?
Yeah, and I will start by saying, and I see there's a question in the chat about informed consent. So starting the informed consent process at the beginning of the trial, in our own case, it takes at least an hour and a half to 2 hours to get through those consent forms, right? Because there's a lot of information at the beginning of the trial that we can share, right? So we can share in the beginning of the trial, this is the information from the animal studies. This is the safety concerns that they had in the animal studies. This is how we're going to monitor. This is what we're going to look for. This is the reason why we're doing this set of labs or that set of labs. So the informed consent process is very important and does allow us to share a lot of what we do know, right? So that is key at the beginning of the trial. As a trial evolves, we've been in this situation, or where you hear about someone having an adverse event, or you get a notice from the other people, the members of the trial sites or from the sponsor, saying there was an adverse event of somebody else in a different site, for example, right? So the sponsor is responsible for determining if something is related, going back to my example of liver failure. If it's not related to the study drug, right? For example, maybe somebody took a lot of Tylenol or was on a seizure med that caused some liver challenges, right? If it's not related to what's being studied, then we may not hear about it at an individual site because it's not considered related to study drug. If there's something that is related to study drug, then we are made aware and do have an obligation and to share it with the families. I think what you're asking though is in that time where something might not happen before everyone determines, is it related, is it not related? So what I say to families, and I reassure them that there is a data safety monitoring board for each trial that is it's not the sponsor or the pharmaceutical company is not. They're looking at their own data, but they are not this third party. A third party is reviewing the data in these scheduled windows and makes the decision: is safety a concern, or can the study proceed, or do we need modifications? So I try to reassure families that even though we have these confidentiality agreements and we can't always say X, Y, Z, if there's information that is coming through, we are permitted to share it. And for the information, as we're waiting for decisions to be made, I reassure them that there is a data safety monitoring board that is reviewing regularly clinical trial data to make sure that safety is first and foremost.
Okay, yeah, thank you for that. Dr. Wong, what are your thoughts on how trial sponsors and researchers can enhance that transparency with families in helping them make that decision? And better understand why there are going to be certain requirements in this trial, and and help them in that informed consent period make these strong decisions about participation.
Thank you. Hopefully my audio is okay. I've been kind of hopping in and out, so if I start sounding robotic, just let me know. I think it's really important. I think Dr. Lau and Dr. Waldman have already mentioned how important the conversation needs to be before study entry. And so just being upfront about what we know, what we don't know, what the study is hoping to do, and just being realistic. I mean, all of us go into doing clinical trials because there is an idea that it helps. There's preclinical studies that show that it helps. Perhaps there have been some patients treated, but this is not like a cholesterol lowering medication where there are thousands of patients per study. There are probably not going to be more than 10, 20. I mean, a rare disease clinical trial that enrolls 100 people is considered a big study. And the important thing about that is there's a lot that we don't know. And so just, having an open conversation at the very beginning about what to expect, why we're doing things a certain way. I mean, I get it. And there are questions about, having to make frequent trips and, especially if your child has disability, that's not a walk in the park. That's not easy, but there's a reason it's designed that way. And oftentimes it's not the company that wants it that way. It's the FDA that wants it that way to assess things like safety, to assess things like efficacy. So it is a huge commitment. I make sure that whenever anybody does sign that informed consent, that I acknowledge just what a huge step that is, and how appreciative me personally, and I think the whole rare disease community is, for stepping up.
Yeah.
Answers your question.
Yeah, thank you for that. So when my family, when we chose for our daughter to participate in a clinical trial, we live on the East Coast. We were traveling back and forth to the West Coast for a long time, for a period of 5 years. And it was a lot of travel. How can we, there's a question in the chat about how we can make these considerations. Families have to realize that our sponsor was very generous in helping us with the indirect cost, the cost of participation, where the sponsor took care of a lot of that. There are still costs that happen. When you're traveling and you're gone a week, either someone's going to have to take care of your pet or you're going to board that pet. That's not included. That's not the kind of things, but you have to be prepared for those kinds of things. How do you talk with families about these indirect costs, and so that they can make these considerations and feel like it's equitable. They have the chance to also participate.
So I was frantically writing down some notes from the chat and stuff. And Christelle, thank you for your the ask about travel support. So we enroll any patient who is interested and eligible. We do talk about the fact that there are all these costs, but many, not all, many sponsors will cover costs of flights, hotels, food to an extent. So I think some of the practical things that we were just discussing. So we pushed back when a sponsor said 1 parent, 1 child. We said, oh, no. 2 parents for 1 child. If the family wants to bring 2 parents, and especially at CHOP, we often require 2 parents for the initial consent process, right? So we push back, but it would be extremely, extremely helpful if the patient advocacy groups also push back. So if you're part of a patient advocacy group and the sponsor is developing a trial for your disease, it would be very helpful if you said, you need to cover the cost of 2 parents and 1 adult, 2 parents and 1 child, for example. Or if there's a sibling, you might say, we need the whole family to be covered because we can't have a babysitter or leave them for several days. Anyway, The other thing, Christelle, is that we prioritize people no matter what their financial situation is, right? So because all of the, many of the big costs are covered, right, in terms of flights, hotels, and food, but sometimes they'll push back and say, oh, you only get $60 a day for food, or you only get $250 for a hotel. And in some of our cities, that's not realistic either, especially different times of year and those types of things. So the role of the patient advocacy groups are to strongly advocate for support. We just had a sponsor tell us that they were going to pay $70 for travel expenses. I said, that's not going to work. Then you're only taking somebody that's in my geographic area, right? So those types of things. The last thing that I will mention is they will often say that you get your travel expenses covered, but you also can get $100 as compensation for the visit, replacing your loss of wages from work. However, that might be $100 over 3 days. And so that's not a lot of money to really cover your lost wages. And the last thing to mention is tax implications. So if you are reimbursed, or I'm sorry, if you're giving compensation of $100 a visit and you make, and there's more than 6 visits in a calendar year, so you make more than $600, you get a W-2 form. I think that's the right, like you get a tax implication form. So there's lots of things here, and we work with our families to try to be transparent about those costs and what the sponsor is willing to pay. But I also think it would be helpful if all the patient advocacy groups band together and tell IRBs and sponsors, you know, maybe it should be $250 a day or $100 a day, trying to push back a little bit on what the financial cost is to a family.
Yeah. Thanks.
I'd like to, if I could, from both ends of it. I've run trials where I had to relocate patients for up to a year. And that was fortunate that sponsor could afford it. So that's the other aspect of this, is that, depending on the sponsor, we do see certain sponsors not able to support that. And that's an ethical question of whether they should embark on that. But for our patients, we need to understand that well ahead, right? I agree with Dr. Waldman about equitable access, right? So why do we have only a few sites? Well, in a phase 1, as you know, we only treat a very few number of patients, and we can't put a site in every region. Hopefully by phase 3, when we have an idea that this is working, we can expand. But there are those restrictions initially, both cost, but also because we're only testing in a few. But then you do need that adequate support. And absolutely, in the trials that I'm designing now, we think about this well before we open a site, right? Especially in my area of pediatric neurodegenerative, to caregivers being able to travel. These are common things that should be advocated for. And we need to partner with the patient advocacy community very early. And we do that where I'm working now. So it's really critical to understand that whole family dynamic when we're thinking about a clinical trial. Is it feasible, especially in a gene therapy trial where, the cadence of those visits initially are so frequent that you might have to relocate for up to 3 or 4 months, even if it's too long, right? You can't go back and forth. And so that has to be taken into consideration by the sponsor. And hopefully when you're choosing that as a PI, that you're having that conversation very early. And I know that we are, because I'm having those conversations now as a drug developer on this side of things. So all of that needs to put into action, but there are limitations, right? Some of these smaller startup companies don't have that ability. And that's a thought there. What's happening there? So again, there's some limitations. We can't also do too much reimbursement because that's an inducement, right? So we are, as a sponsor, we can't pay you to be in a trial, right? So it's a very fine line. So we can't offset wages if you're, of a certain amount. But it should be well thought and discussed before you enter the study, right? The impact, not just the financial, but the psychosocial impact as well, right? And to the whole family unit. So we could go on and on about that, but I've been on both sides of this, and it's very important to think about the whole family when we're entering a trial.
So there are some fascinating things that are coming up in the chat that I just wanted to related to this. So a couple of things. So first of all, one of the ways that a pharmaceutical company sometimes suggest that they could offset some costs is to say, well, your visit is part of clinical care, right? So they'll say, well, if you're coming to the doctor anyway for a clinical visit, we'll use the clinical visit for research. The reason that doesn't work is because of some of the things that Sarah suggested on an international way is that, so if someone's coming to me, then I have to bill it to insurance or I have to bill it to the patient, right? And so we're having a lot of issues with Medicaid and crossing state lines. So if you live somewhere and you're going to come to CHOP and being part of the trial bills things to clinical care because you would get an MRI anyway, or you would see me anyway. If Medicaid is then going to deny that visit, then that causes a lot more cost to the family. And so with international patients, this is a big deal because they don't have US based insurance. And it seems like the question you're asking is, I heard the MLD trial, a patient received an outrageous bill because they didn't have health insurance, right? So the international patients are a little bit different. But if things are being billed to clinical care, then that international patient is going to be billed for the clinical pieces of that study. So one thing that is not typically in consent forms, but something that you should ask as a patient is which procedures are being billed to clinical care, because that's going to be insurance or out of pocket. I hope that addresses very quickly some of the things in the chat.
Critical. And when we're designing a study, again, from a sponsor side, we are very clear about that. We can't expect patients to use in many scenarios for reimbursement. Some of these procedures are experimental. They're not standard of care. They're absolutely not standard of care. So it's actually on us to take that risk on that, we're going to pay for that procedure and all the risk that is associated. So just again, if there's an adverse event that comes from the drug, the sponsor is actually on the hook for that. So that medical care. So we talk about this before when I was the PI with my patients about, what happens, especially in the US where healthcare is not universal. So they can't just assume that going to the ER for an adverse event, is that cared for by the sponsor or is it by the patient, right? The family. And oftentimes it is by the sponsor if it's related, but it's very scary to be in that position. Absolutely. And that's why, again, this goes into the informed consent. It's not just about the drug. It's about everything related to the trial and understanding, financial impact as well as the health impact of potential adverse events.
Right. I've said so many times, you can't say to a family enough how important that period of informed consent is. And do they sometimes have people who come in and to listen with them to hear? Sometimes when you're at a period of anxiety, you don't catch everything. So do families typically have someone who listens with them?
Encouraged. Sorry. Go ahead, Dr. Wong. I know you have a delay there. Go ahead.
No, it's all good. I do think, and I mentioned to many people that, having an extra pair, even 2 extra pairs of ears in the room are really important. Sharon, you mentioned, when you're the parent, you're stressed out, you're anxious. You may not internalize everything that the doctor is describing to you. And so having somebody who is, on your side, but perhaps maybe not as, emotionally affected that day is really important. There are a lot of questions in here about informed consent. And I think that's actually really important because it's not just signing on the dotted line. I think, personally at my institution, I'm the one that gets informed consent because I'm what's called the principal investigator. Ultimately, the buck stops with me. I take full responsibility for this over time. Sitting down. This question is about appearance. Children in language that they can understand sort of what is going to happen to them. They can say no, but ultimately it is the parent's choice whether or not to enroll them. But I think making sure that everybody, patient included, if possible, understands what's going to happen is really important. Sorry, my video keeps cutting out.
We did hear. So there's a question that just came in a few minutes ago. And Dr. Wong, you may have, your audio was cutting out a little too, so you may have covered this. The question is, what are the key challenges in obtaining informed consent from pediatric patients and their guardians, parents making those decisions on behalf of their children? And how does the FDA oversee the informed consent process in clinical trials? We may have covered that. Is there anything else you want to add to that question?
Yeah, so in terms of technical oversight, so every now and then the sponsor will send somebody over to our center and review all the documentation. They have a high interest in making sure that everything is documented properly, because when time comes for them to apply, hopefully for approval of their medicine, they need to make sure that essentially all the I's are dotted and T's are crossed and things were done in a proper fashion. Ultimately, the FDA has the authority to audit all the paperwork. If there were some questions, somebody is going to come from the FDA to look over everything. And so the sponsor needs to make sure, long before there is the potential for an audit, that everything is done properly. And so, yes, there's definitely oversight on multiple levels to make sure consent happens properly.
Right. Okay. All right. Thank you for that. Let's transition to talking about the design of clinical trial protocols for cell and gene therapy. As we've heard earlier this morning, in context of cell and gene therapy for rare diseases, placebo-controlled trials pose challenges due to ethical concerns. There are small patient populations, very small populations often, and the progressive nature of many of these diseases. Many in our audience might even consider the FDA's reliance on placebo-controlled trials for small populations to be, might call it unfair or maybe even unethical. So Dr. Wong, I appreciated your comments in the video this morning regarding ethical study designs. What ethical factors should be considered when choosing between a placebo-controlled trial and an external control arm?
Well, Dr. Lau had mentioned before, first do no harm. If you have a study design that uses a placebo arm that causes harm to the people in the placebo arm, that's not an ethical study design. So I think especially in the field of rare diseases, physicians and their sponsors use a lot of consideration and thought to ensure that anybody in the study who enters is not going to, become harmed by perhaps being a control group. If it's not ethical to have a control group, there have been creative study designs that, allow for the possibility of all participants to eventually receive the study medication. And so it's a very thought out process to ensure that nobody doesn't get the medicine, and anybody who participates has the possibility of receiving.
I'd like to add to that. I think some of the key features, you could see a placebo maybe in an organ that regenerates, right? So there are genetic disorders that are rare that can regenerate. But in CNS disease, for example, as a neurologist, right, this is irreversible damage that can occur while a patient is sitting in an observational arm, even if it's not placebo, even if it's an observational run-in. That is also ethically challenging, because as time goes on, you're losing skills and then you can't get them back. And it's really important to educate the regulators to that. You know, the gold standard is to compare to a double-blinded randomized controlled trial. But in rare disease with end organ damage that's irreversible, whether it's the kidney or the brain, it becomes ethically challenging to continue designing these trials, especially once you have some kind of inkling of efficacy. And that efficacy, I think, Dr. Wong, you mentioned accelerated approval. This is where that comes in, is where we have a biomarker even suggesting that we have target engagement. It becomes very challenging to see continued designs with observational run-ins where you're waiting for 20% loss of a cognitive skill before you roll in. So that's where we need to be, as Dr. Wong said, creative in our study designs and understanding the overall disease progression. So I think we have the ability to rely on external comparators, but that requires an engagement of the community to gather together that natural history. Dr. Wallman, you mentioned natural history data. That is so critical. And that natural history data has to be contemporaneous, right? It can't be that historical that you mentioned as standard of care has changed. So the rare disease community needs to really come together. And we do see that in certain disease states coming together to help with that natural history comparator so that we can reduce that reliance on those placebo or observational trials. So this is an area for me as a PI that I push back against. And now as a drug developer, I'm trying to come up with creative designs with our experts in the field. And Dr. Wallman, would you like to comment as well?
Yeah, I just want to make sure that as a blanket statement, I do not feel placebo is unethical. I feel that these trials should have a placebo design. However, what goes into that is the time, as Dr. Lau was just saying, the time to regression or loss, right? So if you're designing a trial and it is more slowly progressive, so that you have some time to intervene and see whether or not something is going to be different, that's one thing. As opposed to if it was a disease where you have a rapid loss of skills, that's where it gets a little bit tricky to have a placebo-controlled design. So it goes back to what is your natural history, and is 3 months, 6 months, or 1 year, or a delay to getting treatment going to have a negative effect down the line. This is also why the goal is to enroll the fewest patients, right? Because even those that are in a placebo arm have perhaps some additional risk. So you want the fewest people exposed to the study drug and the fewest people in the placebo arm to try to balance out those measures.
Okay. So you bring up the fewest people in a trial. So families often struggle when their child doesn't meet the inclusion criteria for a trial. And often because they're past a certain stage of the disease. So how do you determine inclusion criteria for these trials, and why is it essential? And why is it hard once you've set those protocols? What do we as patients need to understand about how those are developed?
And did you want me to take that one?
Yeah, that'd be great. Yeah. That'd be great.
And then everyone can jump in too. So it's important to know that the sponsor sets the inclusion criteria, right? Not the individual sites or the investigators. So it comes from above us, right? It comes from the sponsor. And the sponsor says, these are the people we think, based on the natural history, are the things that should be enrolled. And sometimes it's age, and sometimes it's ability, and these different things. Because at the end of the day, they're trying to measure a change in one particular aspect, right? And so they say, okay, well, you have to be ambulatory because our key measure is in a timed walk. So if you're not ambulatory, then you're not going to be able to contribute data. So it's a lot of numbers and statistics, right? And so it's not the investigators. It's very difficult for a family to say, okay, well, you missed it by this much or this many months because you aged out. These are very difficult things, but those inclusion criteria are set. And the reason they're set is because they think those are the people most likely to give us the outcome that they are looking at. Not that they want, sorry. It's not the outcome they want, but the outcome that they're trying to measure. And so they're trying to have a very narrow window of time so that they can also see the effects. So for the one last thing I'll say, let's say, and this is a made up example, but something in a clinical disease doesn't occur until you're 5. Well, you're not going to enroll all babies, and you're not going to enroll 10-year-olds. You need to enroll maybe between 4 and 5 to see if you get to that measurement. And that's a little bit of it, I was going to give a Philadelphia sports example, but couldn't think of one fast enough. And, anyway, I don't know if Dr. Lau or Dr. Wong have any other comments on that.
As a sponsor side or the PI side, enriching for a population to detect change is exactly what you're getting at, Dr. Waldman, right? So we can't run trials for 5-10 years. We might follow patients 5 or 10 years, but that pivotal trial, we do want to detect the change within a 1 to 2 year time frame in general, right? So if we don't have a biomarker, which again, that's a whole other path that is potentially very powerful for rare disease, but not every disease has a biomarker. So looking for clinical change within a 1 to 2 time period, sometimes, unfortunately, we do have to enrich for a certain population that's more homogeneous, right? So if we know a specific genotype, we know a specific type of the disease progression, like we talked about earlier, Dr. Wallman, with the rapidity of it, right? That might change what you're looking for in a 1 to 2 year period, and then hopefully expand to other populations. And the other point goes, not every endpoint is relevant to all patients, right? So we also struggle with, like for example, cognition might not be relevant in a patient who's already advanced in their disease, in a neurologic disease. You might need to look at other parameters, but your pivotal trial or your first trial might look at something that's this particular endpoint. And again, it goes back to being creative. That's part of the reason why I'm in rare disease, is that we have to think about endpoint that's relevant to the age or stage of the disease, if you're thinking about neurodegenerative diseases, but what's relevant to that patient population as well, especially if it's multisystemic. There might be multiple symptoms. So you might see that initially in phase 1, we're testing for safety. It's a broader population. You might see it focus in, and hopefully we get a signal. And that tells us that we should move forward. And hopefully we can get access through, other studies. But that's the idea. It's not ideal. We don't want to exclude. I don't want to exclude my patients, but we do need to see a change within a time frame. Dr. Wong, do you want
to? I don't think that the two of you put it really well, that I don't really think I have anything more to add. That's great.
Yeah, that was great. So we've had a couple of questions come in. There are questions about trial design, such as phase 1, 2. What is a pivotal trial? And in our recordings this morning, that was covered pretty. The different phases were covered, but explain more. Go in a little more depth, Dr. Wong, about a pivotal trial.
So a pivotal trial is essentially the big study. It's kind of like the make or break. So you want to enroll a reasonable number of patients to really assess efficacy over a reasonable period of time. At the end of sort of the defined period of time for the study, all the data is compiled and then submitted in the United States to the FDA in what's called a new drug application or a biologics license application. Whatever it is, it's basically an application to the FDA to approve the product. And they have to review all the data, all the safety, and then issue a decision as to whether or not the medicine can be prescribed for a larger number of patients, a broader number, and not in the context of a clinical trial. That's why it's called pivotal, because it's like, the really important phase in getting that medicine potentially approved for all patients.
It's where we all want
hope that answers the question.
Yeah, it is.
Yes.
So I'm going to jump back a little in design, working towards that pivotal trial. So dose escalation is a really careful stepwise process. And gene therapy trials often start with that lowest possible dose. First of all, how do researchers determine that minimally efficacious dose? Anyone? Dr. Long, can you?
Look at you, Heather.
That's right. I'm in it right now, and it's been a wonderful education process on the industry side to see where, right, as a formal investigator, where did they come up with the human translation of dose, right? And that really is informed by your preclinical studies. Hopefully you have an adequate animal model that allows you to see some safety, right, to de-risk that jump to human, first in human, but it's also efficacy. And so what's critical is that, again, we want to move forward to the clinic with a minimally efficacious dose. What does that mean? We need to show something in the preclinical models that there is some kind of efficacy at some feature of the disease. Now, animals are not always going to recapitulate every feature of the disease, but hopefully the key features that are relevant. So you want to understand the dose from both safety, but also do we see some efficacy and function, for example, in a mouse model, and then that will translate to the human. So we want to go in with a dose that it's, again, talking ethics. It's ethically challenged, especially with a gene therapy, where it's not a recurrent like enzyme replacement, where you can do a dose adjustment in ERT, right? Or you can do a dose adjustment with small molecule. But gene therapy, right now, it's a one and done. Right now, it's one dose. So you want to go in with an inkling that dose is not just safe, but have some efficacy. But again, we have to approach it, like you said, very delicately, because again, first do no harm. We don't have all the risk defined in a human. Once we go to humans, there's more, the differences that impact that humans. You start to see other risk factors. So you have to balance risk with efficacy, right? So we can't go in straight to high. And again, creative design, sometimes if we know more about the disease or more about the class effects, for example, AAV gene therapy, you might see us being more comfortable starting a little higher because it's the same platform and we understand biodistribution. So there's a lot of thought, is the point. A lot of thought goes in to try to de-risk that, to go in for that first dose of some efficacy and then try to push that forward. But that's really hard because we know we can't go back and dose those children or adults in those lower cohorts. Dr. Waldman?
So how much time do we have for the panel?
We have about a little more than 10 minutes left.
Okay. Because there were some things in the chat that I wanted to make sure that we got to. I don't know if you want, I mean, I don't think I have anything to add, but I think there's a lot of questions in terms of these dose escalation. Maybe we pivot a little bit into the question about these rolling basis. Right. So if you have a trial and the pivotal trial is more considered the phase 3, but that's not a one-to-one correlation there. But if you have a trial that starts with a lower dose and then moves to a higher dose, and there's rolling patients that come into these trials, well, then, the lower dose gets first access. So you get earlier treatment, but then the patient that might have to wait 6 months or a year for a higher dose, maybe that's the dose that ultimately ends up being the more effective. It's not always clear. These are really challenging concepts for families and for patients and for drug developers, because for a family, you want to get in right away, but you also want to minimize your own risk, and you want the drug that's going to be the most effective. So it's challenging. And I will say, trying to get patients from different areas of the world into these clinical trial sites, it does become very difficult in terms of who gets to go first. And sometimes, and this is done a little bit differently from drug developers, sometimes there's like a central process where they will keep track of patients at different sites, and they say, okay, Dr. Wong, your patient's ready to go next, but Dr. Waldman, your patient needs to wait 6 months or a year or until the next review. Like, there are challenges, and they're not always as cleanly defined as we would like, because maybe we'll pick on my patient. Maybe my patient failed the screening. So then they call Dr. Wong and say, you're up. Dr. Waldman has to wait to rescreen. So these are fluid situations, which can be very difficult for families as well.
That's right. And families talk about it. I know that, I'll get texts from families saying, how do I make sure that I'm number 3 or number 4? Well, you can't.
Yeah. Yes.
If you are invited into the trial, if your application's accepted, you take the spot that works best for the sponsor, the researchers. And I'm looking here trying to compare questions and what's in the
Sharon, while you take a peek through the questions, there was one in the chat that I wanted to jump on.
Go for it.
Matt, a little while ago, and I hope you're still on, you asked a great question about, I'm telling people not to post on social media, which I hope Dr. Law and Dr. Wong agree with. So I'm saying don't post, but then you have a really good point that your voice needs to be heard. And I would say this, your voice should be heard, not on social media. I still believe that that's not the appropriate place, but to the drug developer. So the drug developer, the sponsors, have a patient advocacy group. So you can't talk directly, for example, to Dr. Lau. She's on a side that she should not know who the patients are. She may know where they're coming from, but she should not be able to identify a patient by name or by age, or age you would. But the drug developers have patient advocacy groups. So if you're in a clinical trial, you can reach out to the patient advocacy group. And Dr. Law, if you disagree, please jump in. If you say, oh, I'm concerned about this, or I'm concerned about that, or I want to make sure my voice is heard, that's one avenue. And then the patient advocacy groups, in general, they may set up sort of a confidential thing so they can advocate for all patients, right? So this is something where a patient advocacy group should also work with the drug developer to say, okay, how do we set up potentially a confidential feedback or something so that we can collect the statements and then relay them back to the sponsor. So that way, there's protecting people's privacy. It's like, oh, it's not the person from Texas that's complaining about XYZ, or you're sharing XYZ. I don't mean complaining, but there needs to be these things. And that's where I think the patient advocacy group and the sponsor should try to figure out how to work in that way. But Dr. Law, do you have other suggestions of how I'm not silencing participants, but making sure their voice is heard?
I mean, the other way is directly, because I can meet with the PIs. So there is through your physician is your advocate as well. Your principal investigator is your advocate, and there are infrastructures that you can report also to the IRB for not just oversight of the sponsor, but I'm going to put the PI because I used to be on that too. The PI is also their conduct. IRB, if there is a problem or issue with the site as well. So institutional review boards. The protection of human of subjects is one of the foundations of our research community here. We need to protect. We have all of us are trained, whatever function you are, from your coordinator up to your PI to your sponsor. We're all trained for the protections to make sure that we advocate and keep safe our patients who are in the trial. So there are multiple avenues to get your voice heard. I agree with you. Social media can be harmful, but I do understand, though, that families, it can be an isolating experience, especially if you're in a placebo-controlled trial that I've run. You might feel isolated. There are, again, creative ways that industry sponsors can work with families to get psychosocial support, or with your university that you're working with, if there's psychosocial support, because that community is so important, and you don't want to feel isolated. You don't want to feel silenced. So even if it's going well, it's still, my patients have a burden of they're scared. Are they on placebo, or are they, where can they share? And if they can't share with their community, it's very hard. So that has to be thought out with the institution as well. So there's a couple different avenues, because we don't want you to feel silenced. We want you to feel that you can talk to your PI, you can reach out to the industry sponsor, you can reach out to the institution. So absolutely. I mean, there's a lot of wonderful questions. I'm not sure where else you want to go. If there's other choices here.
And I want to make the point that all of you have stated very clearly how important it is for patient advocacy groups to be at the table in all of this. We're not separate. Gosh, there are some really great questions here.
Sharon, on burden to families, and how do we, so as I said, in the initial phase of like a gene therapy, because we're talking about cell and gene therapy here, you're going to come in more frequently. But we're committed to following you up for a long time for the regulators, but also just for our own long-term safety and long-term efficacy, because we may not see the full benefit within 1 to 2 years. We may not see all the long-term side effects. And so you're signing up not just for the pivotal trial, but you're signing up to be followed, especially in gene therapy. I ran a gene editing trial as a PI. 25 years was the commitment to follow that patient, and I've passed that baton to the next PI. So but it's not as frequent. So how do we reduce burden? We have the ability to do telehealth visits now, the ability to do phone calls. You'll see the cadence of the visits get less frequent. So in one of the trials that we're designing now, or that I've run, Dr. Wong, you've run for disease monitoring programs, or yearly visits, right? So really and truly is to manage expectations, not only of that first trial you're signing up for, but what does it look like in gene therapy that, we want to see you 5-10 years from now? And how do we do that? We try to absolutely reduce the burden. I think that was a question that came out here. Mm-hmm. So you'll see more yearly visits, telehealth, and you're going to reduce the number of assessments, right? We don't have to do every single lab that we did in the beginning, right? Now we understand it better. We're going to focus in on the key safety, the key efficacy to follow over time. Dr. Wong?
Dr. Wong, do you want to have a chance to say? I have a lot to say.
Yeah. So I actually, Mary has a really good question. Yeah. This right to try.
Yeah.
I was going to say, final 5 minutes.
Open up a large can of worms. Okay, so we have time. Great.
Yeah, we got 5
so I know
minutes.
that there are some states that have this right to try where, if you are really out of options, that, you have the ability to sort of try a investigational medication. Cell and gene therapy is a lot more challenging with regards to right to try, because oftentimes there's limited amounts of gene therapy vector. So the sponsor of the company manufactures a finite amount of vector, and it's precious. It is not inexpensive to make, and so they have to make some tough decisions about how to portion out the vector. And especially in the field of rare disease, for every patient that you potentially do a compassionate access for a right to try, that actually removes some of that resource away that could be used for a clinical trial that is actually going to determine safety and efficacy. I know for right to try, sometimes you can get safety data out of.
So while we're losing his audio, just to jump in. Mm-hmm. So my other understanding, and I would love for Dr. Lau to weigh in, my understanding is that for a lot of the pharmaceutical companies, right to try or expanded access, and maybe there's a difference, and Dr. Lau, you can talk about it, but some of these don't come into play until there is enough safety and efficacy data to prove that they work. So if you're in the middle of a clinical trial and there's some safety data, if you still don't have the efficacy data because you're waiting for that to the end of the trial, right to try or expanded access is not an option, right? So I think most of the time where expanded access or right to try could come into play is after the trial ends, while you're waiting to go to the FDA. If you have safety and efficacy data, then that might open that conversation. But right to try or expanded access, just because a drug is made, to Dr. Wong's point, doesn't mean it's available. And to my point, doesn't mean that it's met the criteria in order to give it to patients.
Right. Yeah, I think you have to go back to the definitions of eligible and that it's safe. Again, first, do no harm. I can't. You can foresee that right now there's exclusionary criteria for AAV titers, right, and certain delivery methods for this. So we can't just give drug out there and then have it uncontrolled, delivered by a physician. To gene therapy is highly, needs to be highly monitored and given safely, and you're still there's eligibility here, right? So, and often the right to try is outside of a study. We need to understand in rare disease, every single patient that receives therapy, we need to know how they're doing. We need to understand. So we have to study it and measure it, not just from efficacy, but from safety. And you're absolutely right. It becomes less, it's not really appropriate before we understand efficacy and the safety. And that period between approval right now, it's very difficult, right, to get access to drug. There may not be access to drug, right? So it takes 18 to 24 months to do a batch of gene therapy. Maybe, again, that's not my area of expertise, but it's not like it's automatically produced. So there's so many factors that go into it that we want to give access as soon as possible. And so we're trying to do things in parallel so that, it's not that we want to bar patients, but we need to study it. This is a very, because if an adverse event occurs, and it could actually just lead to the entire program halting if this is given and there's safety issues. So it's a very delicate place to be in. But hopefully, with, again, pathways that are accelerated, that we're trying to get faster with good trial design to get it out there. So it's going from that pivotal trial to BLA to approval. There are ways that we're trying to accelerate that path as fast as possible.
That's right. We have so many more questions that we could discuss. It is truly complex to develop study designs for cell and gene therapy. But I've learned a lot from the three of you, both through the recordings and through this session. I wish we had another hour to talk so that we can get to everybody's questions. But as we close this session, I do want to thank you for the time and effort and sharing of expertise with all of us this morning that you've put into this. Thank you for your thoughtfulness in a very complex conversation. So we're going to take a 5-minute break. Everyone, please come back 11:30 Eastern time because you're not going to want to miss this next session. So thanks to all of you. Thank you.
Thank you, Sharon.
Thanks, everyone.
Thanks.
Welcome back from break. I hope you're ready to settle in for our final, yet very important session for today. Our speakers will explore the critical issue of therapies deemed pre-commercially viable. Dr. Terry Flott, Provost and Dean at UMass Chan Medical School, will bring his passion for addressing this issue, along with Dr. Kara O'Neill, a pediatrician, mother, and Chief Scientific Officer at Cure Sanfilippo Foundation, will help us understand how the MPS community is navigating this challenge. Both speakers will come together for an insightful panel discussion touching on areas of greatest concern for deprioritized programs and, more importantly, the pathways forward. Again, we encourage the audience to ask questions throughout these sessions using the Q&A feature. We will monitor these questions and answer them live during the panel discussion. Thank you.
Hello, and thank you to the wonderful ASGCT team for hosting this inaugural Empowering Patients Summit and for inviting me to be a part of it. Today, I'll share some of the challenges that our community has faced in drug development over the past decade and where we see some incredible opportunity ahead. My name is Kara O'Neill. I'm the Chief Science Officer at Cure Sanfilippo Foundation. I'm a pediatrician by training, but most importantly, mom to Eliza, who is now 15 years old with Sanfilippo type A. To set the stage, Sanfilippo syndrome is an ultra-rare genetically inherited enzyme deficiency. There are 4 subtypes of Sanfilippo that affect different enzymes along the pathway, breaking down the ubiquitous glycosaminoglycan called heparan sulfate. And when these enzymes are inefficient, we are left with partially digested toxic fragments of heparan sulfate that damage cells, affect cell signaling, and cause a lot of inflammation. Now, heparan sulfate is essential to normal neurodevelopment. And so these toxic fragments are particularly damaging to the nervous system. That's why it's common when you hear about Sanfilippo syndrome that there is discussion around the neurodevelopmental and neurobehavioral characteristics of the disease. And that has led to a lot of study around the cognitive impact on children. Children are typically born seemingly normal and have this period of fairly normal neurodevelopment early in life. And it's rare to identify them in this pre-symptomatic period unless they have an older sibling who has already been diagnosed. Instead, children are typically diagnosed around age 4 to 6 at the time when they are experiencing significant symptoms. And at that time, there has already been significant neurological impact on these children. They may be at their peak of neurodevelopment and even starting to lose skills at this time. Now, just to go back, this simple picture really kind of depicts the disease trajectory. This is my daughter, Eliza, at age 4, not long after she was diagnosed. And she was talking up a storm, running all around, playing, and really a vivacious young lady. And our friend Izzy at age 11, not that many years later, had already lost so much, and including her mobility, her communication, her ability to eat by mouth. And she suffered a great deal with movement disorders, dystonia, and seizures before she passed away at age 15. Now, when we think about Sanfilippo and drug development, we know it's helpful to have natural history information. And we're lucky in this regard. We actually do have a great deal of information around the cognitive trajectory, particularly for the 2 most common forms of Sanfilippo. And as you can see here, that by age 5 or 6, we see kids are kind of falling off the curve for Sanfilippo A, and a little bit later, a somewhat shifted curve for Sanfilippo B, maybe around age 8. We really see that happening. And so as a drug developer, you might look at this and say, great, we have a treatment target, cognitive outcomes, let's measure that. We have a clear path forward. And indeed, cognition has been the primary efficacy target for Sanfilippo programs over the past decade. However, this has not resulted in any approved therapies for Sanfilippo syndrome, and we have seen far too many promising therapies lost along the way, as you can see in the red bars. Despite biochemical effect, these trials were either not sustained long enough or perhaps had the right fit of primary endpoint to the population enrolled. And so we were left with the question of, did these treatments really not work? Or were the trials not designed fit for purpose? Did we just have a disease that required a very long runway to demonstrate clinical benefit? And we actually know that that is true, and small biotechs who are operating on a very thin margin with average time periods of trials of 2 years really weren't able to sustain the length of study needed in this disease space to demonstrate clinical benefit. So we lost so many programs along the way. Now remember, if we're going to target cognitive outcomes, we need to intervene very early. And it's difficult to find babies with Sanfilippo. We don't have newborn screening yet. But even if we intervene here, we're going to have to follow those children out for many, many years, up to 5 years of age, up to 8 years of age. And so this is a long commitment. So we need to think about the prevalent population, which are symptomatic children, and what can we measure in those children as well so that we meet the needs of the entire population. We have conducted caregiver preference studies several years ago that informed us of this, and treatment targets that were prioritized by the caregiver community include things around communication, so not just verbal speech, but other ways of communicating, walking and the ability to get around, sleeping at night, eating by mouth, all really important things for quality of life and function. And you see here where that starts to change. So even at that, we know we are going to need to follow these children for a long time, and we need to be planning ahead to include these measures in our studies as we plan them. We are starting to see sponsors, after a lot of advocacy, shift some of their thinking and being creative about how to measure those outcomes in the long term. But I think what it really comes down to is we need to be looking at the biochemical effect that gives us information early on so that that can help predict clinical benefit. And there are 2 gene therapy studies that I'd like to share with you that show what can happen and why one therapy might move forward and one might not. And I kind of consider these sister programs. They were developed by the same scientists, preclinical studies done in the same way, using the same AAV9 vector, delivered in the same way intravenously, manufactured in the same facility, natural history study conducted at the same clinical trial site. So we really kind of have a bit of this platform approach that we understand much more now than we did back then. The first gene therapy for Sanfilippo A began in 2016, and about a year later in 2017, the Type B program started. So let's look at the biomarker data and what we could have gleaned from that then. We see that the low and mid dose cohorts are having very nice dose response reduction in spinal fluid heparan sulfate levels of 52 to 62%. At the time this was presented in 2020, we did not have information on the high dose cohort, but we do expect that would have lowered heparan sulfate more. And the Sanfilippo A program, also very nice dose response to the gene therapy, with the high dose resulting in 65% reduction in spinal fluid heparan sulfate. So when we hold up these 2 programs side by side, we see quite similar biochemical efficacy. Now in 2022, the small biotech that was running both of these programs was no longer able to support their development and tried very hard to find sponsors to take over this program. One sponsor came in and picked up the Sanfilippo A program. That's Ultragenyx, and you may hear about their work during this summit as well. And the Sanfilippo program, unfortunately, did not find a new home and was returned to its academic origins at Nationwide Children's Hospital. Now, as time went on, over the last 2 years, and a lot of advocacy, and we've had the ability as a stakeholder community to share the latest science with FDA around the use of this biomarker. And ultimately, sponsors have reached alignment with FDA, which has opened up the accelerated approval pathway and allowed that runway to extend such that we now have seen some clinical benefit bear out in the Type A program that was able to continue collecting that data. And we see after that 5-year time point, a divergence in the cognitive natural history. So you have to wonder if that pathway had been open just a few years ago, would the Sanfilippo B program have a sponsor and be moving forward? And I think now that we have that pathway open, there's an opportunity ahead of us, and we need to be strengthening the use of accelerated approval in these devastating progressive rare diseases. It works, and it is essential for so many of our children if we ever hope to have them get a therapy. We need to be really thoughtful about our outcome measures, including the patient community in deciding what those are, and really aim to provide the broadest access for children, thinking long-term about what data we need to collect. And then we need to plan for worst case scenarios. How do we run these studies for as long as they need to be run to demonstrate clinical benefit, and plan ahead that if a program needs to be returned to the academic institution, how can that transition happen more smoothly? How can we upfront plan for continued development activities, or at least follow-up of patients after the study ends with the industry sponsor? So I look forward to the discussion that follows. Thank you so much.
Good morning. I'm Dr. Terry Flott from UMass Chan Medical School. I'm pleased to be part of this session and to be presenting my own talk now, Empowering Patients Summit When Innovation Stumbles. The overarching effort we're going to describe is how we will be maintaining access, or how we're going to strive to maintain access for gene therapies, gene and cell therapies that are actually currently, at the current time, commercially non-viable, or what we say is commercially pre-viable, meaning the conditions at the moment might not be suitable for an investor or for-profit entity to pursue these. Nonetheless, we feel committed to maintaining access to these potentially life-saving gene and cell therapies. So I'm going to cover 4 things. First, I'm going to relate an anecdote from our own experience going through an abrupt withdrawal of an IND sponsor, a company that had started a trial and then abruptly withdrew. Then I'm going to talk about the fact that this is a broader trend of therapies that have been abandoned, that have been pursued to a certain level, raising hopes, I think, of families and patients, and then dropped because of lack of market viability. Then I'll talk a little bit about what the American Society of Gene and Cell Therapy, what we, ASGCT, are doing. I didn't mention, but I'm currently the president-elect of ASGCT, and we're very focused on this problem at this time, and we've started a task force, and I'll just share what we're trying to facilitate happening. So we'll start with our own experience. So 2 of my colleagues here at UMass Chan and one of my collaborators at Mass General Hospital, shown on the right of the screen, Drs. Miguel Senya Estevez, Heather Gray Edwards from UMass, and Florian Eichler from Mass General, have had a multi-year program to develop AAV-based gene therapies for Tay-Sachs disease, which is a severe infant neurological disease which otherwise not treated results in a very shortened lifespan. These infants and young children generally only live to around 4 to 6 years of age, really more often 4 years of age or less. With an investigational new drug application of the FDA that did not involve a company, that came through UMass, and with UMass funds and some funds from foundations. In this particular trial, the trial part got some input from Blue Jeans Foundation, but there are longstanding commitments from other foundations, National Tay-Sachs Disease, Cure Tay-Sachs Foundation, Matthew Forbes Romer Foundation. With all of them pulling together, we took this vector treatment that Miguel and Heather had developed, and I was called in to lead the clinical trial effort on this, and a unique trial which involved injecting the vector deep in the brain, which is shown by the little yellow arrows in the upper left on the MRIs. And then over a longer period of time, we were able to see evidence that there was some recovery and some myelin tracts being generated, which are shown in green in the lower right. These were new tracts added. It was far from a curative effort, but it was a first step into patients. There was only enough here to do to treat 2 patients. So, however, because this had gone through various different animal models of Tay-Sachs and Sandhoff disease, and now we had human experience of 2 patients, a company, Axevent at the time, which later changed its name to SIO, licensed the technology and has said they would put some money into doing a phase 1/2 trial to go from these very rudimentary steps where we were giving very small doses, shown down here as the starting dose level on the bottom. They said, well, we'll fund this study to go up 3 more dose levels to really max this out and see if we can see clinical benefits and a greater proportion of restoration of the defect. And not to go through this in too much detail, but the first patient was treated in late January of 2021. But then after having treated the first 5 patients in the middle of April 22, as we were just getting ready to treat the 7th patient, SIO announced very precipitously that they were dropping the study. In fact, they 2 days later laid off many of the staff that were working on it. And so we kind of tried to act decisively as we could and took over control of the IND, which we felt compelled to do, to try to have these patients who had been lined up thinking they might have a chance to participate in the trial to keep going. So we did the last 4 patients, not to get into too many details, with a combination of funds from the university again and from the 4 foundations I mentioned before. These, just to show you, I mean, these are patients who wouldn't have been enrolled. There were 4 of them in total. And some of them, again, not cured, but we saw patients, certainly as we got to the upper levels, who had for a time a really positive response, really something important to build on as they retained critical functions for longer, like the ability to feed and having their seizures managed better. So this is biochemical data. This is really a readout of how much protein the gene therapy was making in the brain. This is a study using cerebrospinal fluid obtained through lumbar punctures or spinal taps. And I put this on here to show that while doing the last 4 patients was, on the one hand, really important to allow us to really fulfill the obligation to all the patients that their volunteering was not in vain. It also, by dropping out precipitously in the middle of a trial, had we stopped there, we would not have been able to see this very clear dose responsiveness, that when we got to the very highest dose, that we would have more of the enzyme. These last patients enabled us to kind of project forward with a newer, better vector that we're hoping to bring to clinic, that there could be restoration of activity into a much higher range, much closer to normal, and actually within a range that we expect we should continue to see some of the clinical improvements that we saw. Now, this is not the only case by far. It's part of a broader trend, and this has actually been published in the peer review literature. So these are just a few, actually 4 of the publications that have shown the stories, or told the stories, of other therapies that have been dropped even after they get to clinical stage, even with some positive findings. In the case of one form of severe combined immune deficiency, SCID, the therapy was actually already approved and being sold commercially in Europe when the company decided it was not a viable a product for the market, even though it was quite effective. So this is an issue. If you look through some of the names here, that this group in the upper left is on the West Coast, primarily of the U.S. It affects individuals in Europe and in Britain. And it's also been talked about in the context of things that we haven't gotten to clinic, but potentially could with a more streamlined process, including CRISPR therapies. Nathan Yingling, along with my 2 colleagues, Drs. Estevez and Gray Edwards, enumerated in a piece that they wrote in Human Gene Therapy about 50 or so examples of trials that were discontinued. This did not even include the SIO, where the effort where the trial was completed, but not with ongoing commitment from a biotech company. There continue to be more such cases, and it's really concerning that so many promising therapies have been taken out of consideration, and really, in some cases, where there is some benefit, even at the investigational stage, denying patients access to these. So, as I said, ASGCT, as U.S. major scientific and biomedical society focused on gene and cell therapy, we've taken a real stand on this, and it in fact is fundamental to our strategic plan. So in our overall strategic plan that came out a little over a year ago, or right around a year ago, we made it clear that our overall central goal, if you look right here in the middle of the bullseye on the right of the diagram that we used to demonstrate our strategic plan, that our overall goal centers on driving universal access to cell and gene therapy. And so that's through many of the things we've been doing all along, helping discovery research, communication and discovery research, innovation to streamline safety and efficacy testing, and trying to facilitate more clinical implementation. This is great, powerful science, and we want it harnessed to give access and impact to patients. So myself, along with Paula Cannon, the current president, Matt Porteous, the vice president, and other members of the executive committee and the board of ASGCT, have set up a task force, which I'm leading, on access. Now, to just state the problem very succinctly, of the 7,000 rare genetic diseases, And there's more than 7,000 rare diseases, but we think about 7,000 of them are single gene diseases treatable with cell and gene therapy. In some cases, their commercial viability is limited by their small market size, and these are the ones we're defining as commercially pre-viable. So myself as an example, many others who've written about their U.S. experience and their European experience are doing certain trials through universities and teaching hospitals, these academic centers. It is the cost of manufacturing and the long duration of the regulatory process really limit at the scale at which we're able to do this outside of the commercial sector. So what we're in our, since on the right, talk about our effort, since our strategic plan makes this clear that this is access is very important for us. As I said, we convened a task force, and we are working with partners to look at potential solutions to this problem, and that might include a regulatory path called a platform approval. It might include having a centralized facility that might have freedom to operate within that particular platform. Those two could be linked. It does not have to be a single central facility. So certainly you would have different ones for different platforms. And so you could imagine, doing this with AAV, with hematopoietic stem cell-based lentivirus gene therapy for diseases like SCID and sickle cell, T-cell therapies like CAR-T and CRISPR therapies. We also, in this effort, as we discuss these other regulatory pathways that we could consider, we're actually looking at other models, because there may be other ways to approach this with the overall goal of, going back to the left side, lower the costs and shorten the time. Shortening the time is crucial because some of our patients really don't have an unlimited amount of time in which to pursue these therapies because of the nature of their disease. In this discussion, the key point is to be able to lower the cost of manufacturing and to shorten the duration of the regulatory process, because for some patients, the window in which the treatment could be effective is quite limited. So this is actually a slide from Dr. Estevez in which he demonstrates the current state of cost, and not to go through every stage of this, but this process basically hands off after the typical NIH-funded lab research in the animal model finishes. And when one goes through the manufacturing piece along with a concurrent natural history study, about $10 million there, $5 to $10 million on the safety testing called GLP toxicology testing. Each step in the clinical trial going forward has major costs. It all adds up to about 80 to 85 million. And one way to look at this is that if an academic institution was able to go all the way through to the end, piecing together funding, that what's shown on the far right, PRV, right next to the capital, the Pediatric Priority Review Voucher, can yield as much as $85 million to plow back into the program. So it more or less just barely breaks even, but with a multi-year investment. So clearly the goal would be to shorten some of this and to cut these costs, because 85 million is a big risk to take when one doesn't know for sure if this $85 million reward after marketing approval will occur. So that is why we're trying to enable natural history studies to be done cheaper. This defines the endpoints for the gene therapy. That would then lower the entry cost. That plus, cutting the costs of the manufacturing would do the same. And as long as there would be a path at the end that the academic institution could have the marketing approval, this potentially could work, or potentially it could go to the end. We get marketing approval to the company. The nonprofit entities break even, and then it's licensed off to a company if the company were to commit to continuing to make it accessible. So that is a tall order to try to organize how that would happen, shorten the regulatory steps with cooperation of the regulatory agencies, and come up with some shared resources that would enable to lower prices. But that effort, we're not alone in the United States in what ASGCT is coordinating. And so in our task force, we're going to be studying the existing efforts. The ones in Europe are united through something called the Agora Alliance. It's really many different institutions contributing to this. One being San Raffaele Telethon Institute for Gene Therapy, offering the SCID gene therapy in Milan, and several SCID gene therapies. Similar therapies offered in London by the Great Ormond Street Hospital, which is the major children's hospital there. Multiple different hospitals, but particularly the hospital clinic in Barcelona in Spain. Spain has a way to allow that final approval, the equivalent of market approval for a hospital, to hospital exemption. And then at that stage, it can be paid for the way other hospital costs are paid. The U.S., there's an effort from Jennifer Doudna, our Nobel laureate, along with Fyodor Urnov at the Innovative Genomics Institute in the Bay Area that's focused on CRISPR. NIH has had efforts through the Office of Rare Disease, and you see them spelled out here, and then several individual academic medical centers at UCLA, at UT Southwestern Dallas, UMass Chan, as I mentioned, and Nationwide Children's as examples. So we don't have the answers all packaged up for this yet, but I just want to make sure that those in the disease and advocacy community knows that this is the highest priority for us, really, and we are determined to move the needle and create avenues for access to be broadly expanded. Thank you very much.
Thank you both for those really great talks. And Dr. Flott, it's very commendable that ASGCT is taking this on as one of the strategic initiatives under your leadership as incoming president, and the task force is really great to see that these efforts are really combining to battle this accessibility of gene therapies. So we do have some questions starting to come into the Q&A. So for Dr. Flott, is the cost and current socioeconomic factors in the gene therapy world the number one reason that companies drop out of these therapies? And is there a financial outcome, a bigger concern for the investors? So could you kind of talk about the financial landscape and why companies are really dropping these programs?
Yeah, well, thank you. And I'll just start. I also want to thank you, Allison and Dr. O'Neill for this session, because we do think it's really important. So what we're talking about here are therapies where the efficacy is either there or potentially there and being developed, and the actual return in sales in the market is not sufficient to attract the attention of for-profit biotech or pharma companies. There are a few different reasons for this, but the one most obvious is that the markets are very small. So even with the unit price being very high, in the millions, 4 million for some, 2 million for others, even with the market price being that high, the numbers are small when you think about the amount of investment that goes into bringing a cell and gene therapy all the way forward to market approval through the BLA stage. The other factor is that in some conditions, what's been seen is that the patients who have the disorder will be treated. And then once you've sort of caught up with the ones that have been treated, the incidence of new cases becomes smaller because you've basically treated the existing pool of patients. So that's why often companies are looking not just at how many total patients are there in the world, but how many new patients do you expect in any period of time. Now, I would say, the cost of goods is part of what drives the price so high, but I think the small market and the long process to get to the clinic are really things that are weighing on that side. There is also, in terms of social economic factors, there are issues related to reimbursement, and in particular, it seems that in the United States, we have a particular problem in that most of these genetic diseases affect children, and most children in the United States, a majority at least, get their health care paid for by Medicaid. Medicaid in the United States is not a single plan. It's 50 plus different plans, one in every state. And so we clearly see differences in Medicaid coverage for gene therapies between states, and it is even more complicated and less likely to happen for someone to have to cross state lines. So let's just say there's a gene therapy that was commercially being given at Nationwide Children's Hospital in Columbus, Ohio, and you're in another state where Medicaid doesn't pay for that therapy. You have a real problem with access, and I will say that both companies and the sites, which are typically teaching hospitals, academic medical centers, both of them, they are really striving in that context to find a way to treat those kids with philanthropic funds and other things. But that is a huge problem. In pursuing that problem, actually, and I don't want to go rattle on for too long, but insurance companies have looked at this. Is there a way this can be done? And there are creative models. The CMMI, the Center for Medicare and Medicaid Innovation, has a pilot study ongoing that was started during the last administration that to sort of look at payment models specifically focusing on sickle cell disease gene therapy access. And private companies have looked at it as well. But in the end, it's what a company gets in terms of margin for a gene therapy makes it hard for them to look at this and say this is the right way for them to spend their company's assets. Now, I don't want to hog up the whole airtime here, but I have a thought on that, but maybe we can come back to that. But that's currently the why, more or less.
Great. Thank you, Dr. Flott. Dr. O'Neill, from the patient advocacy perspective, what are the biggest challenges that families face when a gene therapy trial is deprioritized or canceled?
Yeah, I think a lot of that depends on what was set up ahead of time, what the resources of the clinical site are, or the academic center that was hosting it are. But essentially, patients are going to feel abandoned. They're going to question what happened. Was this not a good treatment? Why did it, so was there a problem with the treatment? Or is there a problem with us? Is there a problem with our disease? Is there a problem with our community? We're already feeling like the other in the world, right? And so why wasn't this carried forward? Why are we being left to the wayside? And then just that general kind of sense from the community of there's a loss of hope there. It's another kind of ding, and also a loss of trust. And so that kind of erodes trust in the system of clinical trials, of industry, and things like that. Well, who's really watching out for us? Does anyone actually have our interest in mind, or is it all serving some other purpose? So I think it raises all of those kind of feelings.
Great, thank you. And I completely agree with that. Very critical trust relationship that needs to happen with the patient advocates, investors, and sponsors.
Allison, I
Yep.
think just to add also, there raises this question of, well, now who's going to follow up? What if something happens to my child that might have been because of that? And what are the safety things, depending on at what point in the process this is dropped? Who's going to be knowledgeable and help me navigate this for my child? So I think that's another kind of more individual, like, healthcare piece that's
Well, that's a
important.
good segue into another question that is about transparency when these programs are deprioritized, and how can the field ensure better communication and accountability to the patient community? For example, patient communities try to move a program forward. They could benefit from having the access to the clinical trial data, but there's currently no incentive in place for the companies to share this. So, could perhaps both of you comment on this lack of transparency and how the providing some of that data and continuing to follow up the patients could help to continue to benefit these programs?
Well, I certainly agree with everything Dr. O'Neill just said. So, well, first of all, I think what needs to be defined here, and I don't know if it would have to come probably from FDA, but it might have to maybe come from IRBs, is that really when the patients begin to participate in the clinical stage of a product, I think there is an inherent or there should be a commitment from whoever has started that effort to follow it up through a specific stage. So I think it would be totally irresponsible for instance, begin a trial program and then stop it before it was intended to stop because it was costing too much money or whatever. And I guess the way I put it is that people who work for-profit companies seem to have a lot of concern about their responsibility to their investors. I think we should define what is their responsibility to the patients in that community if they have started having them volunteer to put their children at risk for such a therapy. There isn't, in my view, there's a moral obligation there. There should be an obligation that is defined. There's some kind of commitment, including the commitment for long-term follow-up, that somebody has to do that. And thankfully, a lot of the academic medical centers that work with companies are willing to do that if they were part of the early development, for sure. But there's some responsibility there that I think needs to be better kind of defined and held to, and that would of course include transparency. Not being honest with people is kind of the worst thing you can do with them, I think, in a situation that's so serious as this.
Yeah, I think there's always a lot of concern around compliance, and we can't tell the community XYZ because of compliance concerns that are not ever really very clear as to what is the actual concern and why can't we talk about XYZ. So I think that's really confusing from the patient side of things. Why can't we know certain things? And around the transparency part, and having access to data later, that commitment to, people enroll in these studies because, yes, there's desperation, they want treatment for their children, but they also know that this is science and we don't know what the outcome's going to be, but we sure as heck want other children and the field to benefit from it. We don't want our participation to be stuck in a closet somewhere, never to be seen again. It has to be generalizable knowledge that comes from this, and that has to be put in that informed consent to enable data sharing from the get-go, and that be the expectation. At a minimum, when the trial ends, patients should be getting back their data. They shouldn't have to be begging for it, knocking down the doors. Here, you're done this study. Here is your medical information. This is what we learned. I mean, there's such a power imbalance between participants in studies and the system. And when you have a deadly, terrible, horrible, rare disease, you are have very little power to even ask for what is right. And so, to your point, Dr. Flatt, the expectation should be there. Where is the requirement to do what is right in that situation?
Yeah, one thing I'd add, if a company has already gone public, they seem focused to the nth degree on what the SEC will let them release, tell some people versus other people, or really harping on the idea of insider trading, instead of recognizing that, you've got patients and family who really depending on those who are doing the trial to be honest with them and to sort of give them something back that is in return for the risk they're taking on, and more hospitalizations in many cases and other things like that. So I think we need to define the patient's rights a little better in this situation.
I agree. Thank you both for that. Okay, another question from the chat. Dr. Flatt, you touched on this briefly in your talk, but there are some programs that are dropped after approval. This question states that this might be due to lack of interest in patients or lack of demand. So what are your thoughts on why these products that were not successful as compared to products that are earlier in the development stage?
Yeah, well, I think there are circumstances, and I think maybe in the chat I'm not looking at it right now, but they mentioned hemophilia. I think there are circumstances where a disease is a very good candidate in terms of proving that it worked, because in hemophilia, measuring the clotting ability in the blood and the factors levels is relatively easy. Compared to looking at neurodevelopmental outcomes, for instance. It's a blood test. So from a standpoint of getting a trial done and proving efficacy, that's easy. But to bring it into the market, it's not only got to be effective in a condition like that, it's got to be substantially more effective than the existing therapy. And these existing therapies for hemophilia have gotten better and better. So I actually think that is really a relatively small proportion of the 7,000 genetic diseases that we talked about. So many of them are diseases where we'll have a biomarker, as in the Sanfilippo case and in the Tay-Sachs case. We'll have a biomarker, and we'll have to look at long-term trajectories on development and neurologic function. Long term, and then we don't have very many controls in our natural history study. And so the outcomes become, the functional outcomes become much less easy, but the treatments in those cases are much more needed than in a disorder like hemophilia where there are other treatments. So I think that's the problem with that. I do think there's uptake problems in diseases that do have a lot of severity in them long-term, like sickle cell, and there are a variety of reasons for that relate to health equity and equity of access and income and various other things like that.
Great. Dr. O'Neill, in your presentation, you talked about how accelerated approval pathway can really be a game changer for some of these programs. Can you both touch on briefly that if there would be a more established accelerated approval pathway or similar pathways like that for gene therapies, how would that change the trajectory of these programs and how many of them succeed?
Well, I think kind of on a real patient level, I think that lets us expose less patients to non-therapeutic doses, so we can have more timely dose escalations. Because right now we are not redosing patients, and those patients give up their opportunity to have a therapeutic treatment, essentially. So I think, safety-wise and ethically, that really helps us move quicker. And I think it could save a lot of programs, honestly. I mean, you have to have a disease where there is a biomarker that the science backs up, obviously. So you have to be starting from that. But Dr. Flatt?
Yeah, I certainly agree. There are two big problems with taking a long time. One problem is if it's a small company, they often talk about what they call their burn rate, which means just keeping people employed in some of these companies for 3 to 5 years is a lot of investment in and of itself. And if that's shorter, then there's less time between when they start basically paying for the operations and when they start getting some revenue back in. So that's part of it. The other part of it, of course, is we're talking about conditions here in these particular examples where, there's a very limited window for intervention. And while it might sound like, oh, we have to go methodically, whatever. Well, if depending on when your baby's born in the developmental process of that therapy, it may be too late by the time it gets out. So I will say it's interesting. FDA has these various ways to get accelerated approval, breakthrough designation, orphan designation, all these. And one thing they did, so it's a little confusing because I think different ones of them came out in different legislative initiatives. But one of the things they did that I hope gets followed up on is they had a pilot program for a couple of diseases called the START pilot, in which they basically were helping people navigate through all of these variations, that can help. So I think they need that, considering the way the different rare disease oriented incentives or pathways are hard to kind of parse out which ones fall into which category or which ones can benefit from more than one.
And a lot of times, folks that decide to enter this space are very, very tiny, under-resourced companies that are just startups. And so they may not have the experience or the staff to navigate all those regulatory things. And the volley and response that happens with our regulatory system with like the letter to the letter response, it's not the same as a conversation. And so that START program is an incredible opportunity to just have that real time. Like, let's just get to the answer and what's the next step.
Great, thank you. So I'm going to have two last questions. The first one is, gene therapies often have a narrow window of clinical effect, particularly in neurodegenerative diseases. How could some of these endpoints be reconsidered to allow for broader therapeutic opportunities that are meaningful for families?
I think
I touched on that a little bit in my talk, and that's kind of a lot of the advocacy that I talk about, because I don't think just because you're not a baby and you already have cognitive impairment that we should say you don't get medical care, you don't get treatment. No, we need to treat our children because that's the right thing to do. But it is finding what is modifiable at that stage and looking across the spectrum and having those appropriate expectations around risk benefit at the time of intervention. And in our case, it's other factors, still many of which are neurological, but just not cognitive in nature.
Yeah, sometimes I think what happens is you have the biomarker, and then you're going to look at different outcomes, and they want you to say, well, we're going to power the study off of this outcome. But after you do the treatment, you find that other aspects of the disease are responding more than the one that you put in. And every disease is different. So I've heard a proposal that, put out multiple different endpoints, and if any one of them is hitting in tandem with the biomarker, maybe that should be enough for some of these rare diseases, because it's just hard to redo the study and say, well, we do it over again and drive off of this outcome versus that one.
Yeah, that regulatory flexibility in these ultra rare diseases is so critical, and this idea of looking at that totality of evidence.
Great. Okay, for the last minute, Dr. Flood, I'm going to let you have the last word on what you think are some of the most promising alternative funding mechanisms or partnership models that could help support the development of rare disease gene therapies.
Well, I'm glad we get to talk on the promising part. So I will just tell you that last week, this task force that we put together got a number of people together, about 50 people in a room for a day and a half, and with another dozen people on Zoom. And we started hearing all of the different ways different people are trying to do this. What they're doing in Europe, which is a little different in UK than in Spain and in Italy. What some people are trying to pioneer this concept of a public benefit corporation. There are innovative concepts around not-for-profits. And I think one of the most encouraging things is that one of the real leaders in support of this initiative is Peter Marks, the head of the Center for Biologics. And FDA wants to help us do this in this way. So I have a lot of hope. I will say this, of those, whatever it was, total 60 some people, everybody agreed that we should be changing to improve access. There was not a single person to say, oh, that's a naive idea. I guess they wouldn't have come. But I always say to everybody who brings this up, I say, if we're smart enough to figure out how to actually do this, we should be smart enough to figure out how to pay for it and make it accessible. It shouldn't be, it's actually shouldn't be as complex as getting an effective therapy in the first place.
Well, I think that is really a perfect ending of the day. I hope that we started our day showing you all some of the ways that we are smart enough to develop a lot of these tools that are now reaching the clinic for cell and gene therapies, how we have used some innovative clinical designs to make successful gene therapy clinical trials. However, we ended talking about how sometimes innovation does stumble and what are some of those roadblocks for accessibility of gene therapies. However, there are some innovative concepts and some new ways to think about how we can continue to move these therapies to the clinic and market approval. That was a perfect segue for tomorrow. Please join us again and have your morning coffee with Dr. Peter Marks from the FDA, followed by learning more about manufacturing, plus Patient Advocate Foundation leaders share their experiences navigating gene therapy development. All of these sessions from today will be available shortly on demand here on Brand Live for the next 30 days, and we will be open for access on YouTube thereafter. We welcome you to follow the instructions on the screen and submit your feedback by completing the event evaluation. And we would really greatly appreciate hearing your thoughts. I'm so thankful to all the presenters and panelists and moderators today, and we really look forward to seeing you tomorrow.
The field of gene and cell therapy is rapidly advancing, with treatments including gene addition, RNA therapy, gene silencing, cell therapy, and gene editing. Some are already FDA approved and available at sites around the world to help specific patient populations, while others are being studied in clinical trials to see whether they are safe and effective at treating or preventing certain disorders. These treatments all aim to target the cause of the disease on a genetic level, and there are a variety of ways to approach this. A gene is made of DNA and contains the instructions for a cell to make a specific protein. While DNA stays in the nucleus, RNA carries these instructions to the cytoplasm where they are used to make proteins. Proteins are the building blocks of how we function, so they make a big impact on our health. First, let's talk about gene addition. A gene variant may be causing not enough of a specific protein to be produced, or the protein produced is not working. Gene addition adds in a properly working gene that has the instructions for the cell to make more of the specific protein. A vector, which is often a virus, delivers the working gene to the cell's nucleus. The viral genes are removed to ensure the vector cannot cause disease and are replaced with the working gene. Because the added therapeutic gene will then reside in the nucleus, it has a greater chance of being permanent and is only administered one time. Gene addition isn't the right approach for all disorders. Another approach that may be explored for treatment is RNA therapy. There are different types of RNA therapeutics because there are so many different ways that RNAs can affect cell functions. For instance, messenger RNA, or mRNA therapy, is designed to produce more of a specific protein, similar to gene addition. However, sometimes gene variants may cause a protein to be made that has a toxic effect, and leads to disease symptoms. In these cases, microRNA, interfering RNA, or ASO therapy can be used to silence the faulty gene so the protein is not made. ASO is a very short sequence of RNA or DNA that can be delivered to cells to change how a protein is produced. These stay in the cell for a limited duration and may need repeated doses to maintain a therapeutic effect. Some RNA-based therapies can be delivered using a viral vector. In this case, the instructions or DNA to make the RNA therapies would be given directly to the cell and would be a one-time treatment. Cell therapy is another approach and can be used to target a specific cell type to treat disease. Here to explain more is Dr. Daniel Bauer.
Cell therapies could be based on blood stem cells or immune cells for disorders like sickle cell anemia or certain forms of cancer. Many cell types have the potential to be modified. Depending on the type of cell therapy, the cells used may be from the person's own body or from a donor.
Genetically modified cell therapy removes a person's own cells from the body. The removed cells are treated by adding the working gene or modifying the affected one. The treated cells are then returned to the body. This type of treatment combines components of gene and cell therapy. Since the cells are provided with the corrected genetic material, the therapy would likely be longer lasting and may be given one time. This is only a snapshot of all the ways gene therapy, RNA therapy, and cell therapy are being applied to help treat different disorders. There are decades of research that experts continue to build on, with new findings and developments happening every day. For more information on these different gene therapy approaches, or if you're curious about gene editing, please visit patienteducation.asgct.org.